The evil, dark dragon blade. A legendary sword, said to have been carved out of the bones of a black dragon.
There, I'm not going near that one. Lifespan, the dimorphism in lifespan tends to be in between, and parental behavior and likelihood of all of those. You look at a number of measures, and by next lecture, we will be looking at some genetics of what monogamous species and tournament species look like, and we're right in the middle. In other words, that explains like 90% of literature, because we're not a classic tournament species and we're not a classic pair bonding one. We are terribly confused in the middle there. And everything about anthropology supports that. Most people on the planet right now are in a form of monogamous relationships, in a culture that demands monogamy. An awful lot of people who are in monogamous relationships in such cultures aren't really in monogamous relationships. Traditionally, most cultures on this planet allowed polygamy. Nonetheless, in most of those polygamous cultures, the majority of individuals were pair bonded and monogamous. You get two different versions of polygamy in different social systems of humans. One is economic polygamy, which is you're basically sitting around and the wealthiest guy in the village is the one who can have the largest number of wives, an enormous skew in reproductive success that's driven by economics. The other type is demographic. You have a culture where, for example, you have a warrior class. Guys spend 10 years as warriors, New York City accent, as warriors. They don't worry, there's no anxiety, but they eventually worry about getting a wife, because by the time they're done being a warrior, they're like 25, and they marry someone who's 13, which is what you see in a lot of traditional cultures that follow that pattern. And at that point, you got a problem, which is an awful lot of those guys have been killed over the course of 10 years of being involved in high levels of aggression and 10 more years of life expectancy to catch up with you. There's a shortage of males. So you see polygamy there driven by demographics, and you see polygamy driven by economics and other types of society. So most cultures on this planet allow, traditionally, before the missionaries got them, most cultures on this planet allow polygamy. Nonetheless, within most polygamous cultures, the majority of people are not polygamous. We have one really confused, screwed up species here, because we are halfway in between in all sorts of these measures. Okay, so what do we have next, which we will pick up on Friday? What we've just started with here is the first case of using all these principles, individual selection, kin selection, reciprocal altruism, to understand all sorts of aspects of behavior. We will then move on to seeing how they explain other aspects of animal behavior, some ones which, if you are behaving for the good of the species circa 1960, there's no explanation at all, because you're doing things like killing other members of your species. And then finally, we will see how this applies to humans and some of the witheringly appropriate.
For more, please visit us at stanford.edu.
let's get going. Various announcements, procedural things. A number of people want more information about grading and what the exams are like, all of that. I think I mentioned 1/3 of the points will come from the midterm, 2/3 from the final. In terms of the style of the midterm is heavily going to be about making sure you got down all the factoids from the first half of the course, that you've got the basics of each of our proverbial buckets. The second half, the final, is all going to be about integration, thinking across the different categories. So just a sense of that. Readings, as they are coming out, the books are not required until the second half of the course. The handout on Monday, I think, said which chapters of the Zebra book you should read. We will shortly get to you which chapters of the Chaos book you should read as well. The readings that are being posted on the courseworks, the downloads of various published papers, those are required. I'm clear on those whether this is a paper you should read all of, if this is one you should read the abstract of. Even if you read all of, do not read it in some obsessive, detail-oriented way. The goal is probably to be able to say in 1 or 2 paragraphs why this paper has something pertinent to say about the topic they fell into. You're not sitting there having to memorize techniques, middle names of the authors, how many animals, anything like that. In terms of that, it probably makes sense to read those after the first lecture of whatever block there is. And hopefully, if I get organized, I'll be able to get you sort of the list of the readings further in advance than 1 week in advance. Nonetheless, you should probably hold off reading it until after the lecture occurs. Let's see, what else? People wanted to get a sense of how long things were going to go. And as we'll see today, the evolution lecture topic will cover 2 classes. Molecular genetics, which is what we'll pick up on Monday. I'm guessing 1 to 1.5. Behavior genetics following that, 1 to 1.5. Ethology 1. Neurobiology, endocrinology, we'll have 1 week devoted to intro to the topics. And again, that's one where this is so important for everybody to be up to speed. Rather than these being in catch-up sections that week, the whole week, Monday, Wednesday, Friday, will be devoted to that with the TAs teaching it. The following week, 3 more lectures, more advanced ones. And depending on proximity to the midterm, there may be a half lecture in there on statistics, or maybe not. So it's going to depend on keeping on schedule. This is a rough approximation. The midterm is going to be a Monday night. You will be responsible for material up to the previous Wednesday, and there'll be lots of review stuff. Take a look at the extended notes being posted. What else? Okay, I think that covers most of the procedural stuff. All of the stuff will get posted as well. So, picking up on the other day, what was happening the other day? Number 1, the trouncing of Darwin inventing evolution, trouncing of survival of the fittest, probably more importantly, trouncing of behavior for the good of the species, group selection type arguments. What we saw was number 1, the rationale for the whole thing. There is a vicious, unfightable logic to why hearts have to be the size they are and kidneys have the filtration rates they have to have
The
in order to
evil,
solve the
dark
challenges
dragon
of
blade.
leaving as many copies of your genes and making sense of the evolution of hearts and kidneys and things like that. Could be the worlds of bioengineers and biomechanics folks with an underlying logic that it's got to be something that increases the number of copies of genes you need. And the whole rationale for Wednesday's lecture today is applying the same sort of logic to behavior. The whole world of just as you can optimize sort of the way one's neck, how long it is if you're a giraffe, you can optimize behavioral strategies. And again, also throwing in that caveat, no animal is sitting there, maybe with the exception of some other apes, sitting there consciously strategizing along those lines when saying, so what would you as this dandelion want to do at that point with this ecological challenge, personifying just to make things easier. Okay, what we've then barreled into were the 3 major building blocks for thinking about the evolution of behavior in the framework of contemporary evolutionary thinking. Number 1, individual selection, passing on as many copies of your own gene to the next generation as possible by way of your own reproducing. the individual selection. A chicken is an egg's way to make another egg. I've now rehearsed that, so I've got that down right. The whole notion of behavior as just being this epiphenomenon in order to do what's needed to get another copy of the genes into the next generation. Building block number 2. Some of the time, the best way to increase the number of genes you pass on to the next generation is to help your relatives do so. Following that logic of Mendelian relatedness, and people in the catch-up section I know went over issues of why is it that you share half your genes with a full sibling, a quarter with a half sibling, etc. So sometimes the way to maximize is by helping out a relative to do so with, again, constraint by this vicious mathematical logic of it depends on how related you are to the relative. And thus you will gladly lay down your life for one identical twin, 2 full siblings, 8 cousins, off you go. So the whole notion there of insight into why social animals the galaxy over are so obsessed with kinship and relatedness, the whole world of who counts as an us, who counts as a them in terms of cooperative behaviors playing out along lines of relatedness. Finally, we saw the third piece, which was reciprocal altruism. You scratch my back, I'll scratch yours. Many hands make the task less scratchy or whatever. And what you see in those cases there is a whole world in which you don't have to be related to have cooperation. And we saw all the domains of that, bringing in the formalization, biomechanics person figuring out how strong a leg bone has to be, a game theorist figuring out within the realm of social behavior when you cooperate, when you don't, what sort of strategies. Game theory of seeing the prisoner's dilemma as the building block of that entire field, and seeing all this strategies worked out by mathematicians and economists and diplomats, and seeing which ones optimize under what circumstance, and then going and looking in the real world and seeing there's all sorts of animals out there that have evolved optimization strategies of when to cooperate and when to defect. And what we'll get to as a huge, huge issue by the time we get to the lectures on aggression, cooperation, etc. is, well, that's great when you've got a cooperative system going. How do they ever start? How do you jumpstart cooperation systems, the evolution of cooperation? That's something we will get to in great detail down the line. Finally, what we shifted to was now saying, great, we've got all these principles in hand here and our big 3 building blocks and all of that. How would it be applied to making sense of animal behavior out in the real world? And we left with the example starting someplace there. Where did it go? Yes. That's it, of us marching through no one individual factoid about some species or other species, know that there's a big difference between the genders in size, or there isn't, know that there's not high levels of aggression in males, know that females always give birth, whatever those traits are as we marched through, using these ideas about individual selection, kin selection, reciprocal altruism, you could march through and logically infer what the social behavior of this particular species was going to be like, and you would be right. We saw, for example, in tournament species where you have high levels of aggression among males, male-male competition for access to females. As a result, males tend to be a lot bigger than females. They are being selected for muscle mass, secondary sexual characteristics, plumage, big sharp canines for slashing the other guy, bringing up this issue of, in a tournament species, who does the male want to mate with? What sort of female are males interested in mating with? The answer being anyone who will mate with them, because there's like no cost involved. All that's involved is no parental behavior, none of that, the cost of sperm, and literally people analyze the relative cost of sperm versus eggs. In tournament species, males are dramatically unchoosy as to who they mate with. In tournament species, males have dramatic variability in reproductive success, 5% accounting for 95% of the matings. Female choice in a tournament species, what does a female want out of a male? She certainly is not going to get good fatherhood out of the guy. All she wants are good genes, because that's all she can hope for from the guy. A whole world of female selectivity for markers of good genes. And as we'll see in the sexual behavior lectures, a whole world of males trying to fake out females across the animal world, suggesting they got better genes than they actually do. So we will come to that. Then, tournament species, do you see female abandonment of kids? Absolutely not, because there's nobody else who's going to take care of them. Coupled with that, you see single births among those species, high levels of aggression amongst the males, low levels of parenting, and males tend to live a lot shorter than females. And what we saw at the other end, the pair-bonded species, was a completely different picture. Males are being careful who they're mating with because the wiring there is you mate, you take care of the kids. High levels of male parenting, females are thus able to abandon them. Females can have multiple births. Males are selected to be as close to females as a male can be, so there's not a big difference in size, there's not a big difference in secondary sexual characteristics, there's not high levels of aggression, there's not big differences in lifespan. Who does a female want to mate with? Someone who is going to be a competent father, thus the whole world of pair-bonded species where males court with rituals of coming up with food and coming up with things of that sort. So the most striking thing about that was, again, going back to the two skulls I brought in, you could know nothing more about that species other than this is an adult female, this is an adult male, and working through the same work, you know a huge amount about the private lives of this species and who's messing around with who in the bushes, and that's merely by applying these principles. And likewise saying, here's a picture of a pair in this species, and you can't tell which one is who by gender. That tells you a whole world of predictability at the other end. So a first example of how much explanatory power you can get out of this. And on the website, I will find all sorts of appropriate pictures of tournament and pair bonding species for you to look at. Okay, so a first realm of looking at animal behavior and seeing how once we got and individual selection, selection, reciprocal altruism, suddenly an awful lot of stuff makes sense. Next domain where that's the case. And this is one that has had a real challenge to the, ooh, isn't nature benign and animals behave for the good of the species viewpoint. This one domain probably proves an individual selection framework more than anything else out there. Okay, back to our inevitable National Geographic specials. And somewhere at the end of whatever the special is about whatever species, at some point they usually get some narrator with some deep baritone voice come in at that stage and say something like, man is the only species that kills for pleasure. Man is the only, and look at how wonderful and benign these rose bushes are. They don't kill, they don't have war, only humans do it. And what occurred by about the mid-70s or so was enough field workers from a different species reporting, hey, wait a second, we're not the only species that kills. And we will see plenty of domains where that makes sense in the lectures to come. But one particular domain really demanded some rethinking about animal behavior, which was as follows. What is one of the universals, whether you start with us and go all the way down to slime molds, it's babies are cute. Everybody likes babies. Babies are adorable. You want to take care of babies. Your eyes dilate as soon as you're around them. And a long-standing notion that what infants, what baby features are about, are, among other things, are merely reduce aggression. And we will see in the ecology lecture something about that. That was the standard soundbite first. And suddenly, you got field biologists coming back from studying various species saying, something's up there because in my species, I see individuals murdering kids all the time. Whoa, what's up with that? Infanticide within a species. Suddenly, not only are humans not the only species that kills, we're like not the only one who goes and kills adorable little baby infants. Suddenly, all sorts of species popping up where there is infanticide. And the huge challenge then became to make sense of this. This first emerged in the 1970s, studies of langur monkeys in India by someone named Sarah Hurdy, reporting this. Soon it was reported among lions and other species as well. And the first obvious response to this was, wait, this can't be because I watch all the wildlife specials. There's something growing. Oh, there's some sort of psychopathology going on. This is not a normal population. This is not a normal population because they live close to humans. There's some disturbance. There's a toxic waste dump somewhere. They're not a normal species because you've got the wrong color socks on as the observer, whatever it is. This is not normal. This is pathological behavior. But long enough time goes by, and it becomes clear in all sorts of species, individuals kill infants. So what is this about? You start to look closely, and there are patterns to it. The first one being that tends to be adult males who kill infants. The next being, you look closely, and it's not random who kills who. It's males killing infants who are most likely to be the offspring of, you guessed it, individual selection, most likely to be the offspring of other males. Competitive strategies for reducing some other guy's reproductive success. So this began to be seen in these species that had infanticide. It was male infanticide of offspring who were of kids who were most likely to be the offspring of other males. But more patterns popped up at that time, which is, well, why don't you see that in any social species where you have competition? And what wound up being clear after a while is there's only a certain pattern that you see in species that have competitive infanticide, which is the average interbirth interval among females is longer than the average tenure of a high-level man. What? Here's what that means. That means you're some low-ranking guy, you've been working out in the gym for years on end, you're finally in a position to boot out the alpha male and take over the whole group, and damn, every single female in the group has a newborn who they're going to be nursing for the next 2.5 years, so they're not going to be ovulating, and they're not going to be ovulating for about 3 years, and you're likely to be alpha for only a year and a half or so. So the length of time on average that you are going to be able to be reproductively active is shorter than the length of time that females in the species nurse kids and thus do not ovulate. And suddenly you have this brutally clear logic that makes perfect sense from everything on Monday, which is go and kill the kids. Go and kill the kids for two reasons. Number one, by killing the offspring of some other male, you are decreasing that individual's reproductive success. This competition is losing as many copies of your genes, etc. Number two, by killing an offspring, the female, by stopping nursing, will soon be ovulating. And thus you see this pattern in langur monkeys, vervet monkeys, paddis monkeys, lions, mountain gorillas, and such, where it's always this structure. Competitive infanticide, a male takes over a breeding group and goes about systematically trying to kill the infants. So we've got an enormous violation there of behaving for the good of the species, and baby animals are cute and suppress aggression and all that sort of thing. The clearest demonstration of that is to look at the fact that one of the species where this goes on by this vicious sort of logic of competition, this is one of the species on Earth that is as close to extinction as you can get, mountain gorillas. There's maybe 600-700 of them are left, and these are just like stunning animals, and they are disappearing because of habitat degradation and human presence and civil wars sweeping through there and all that sort of stuff. But also, they are facing, I think, inevitable extinction in a decade or two, because every now and then a male mountain gorilla will systematically go and kill the infants around in the group by this logic. You can't ask for a much better demonstration of animals not behaving for the good of the species or the group, this individual selection strategy. So immediately we start applying some rules to this. Well, when's a circumstance that a male would come into a group and boot out the previous reproductive male and take over, and where he wouldn't be all that fast to try to kill all the infants in the group? When would a male take over a group and not be all that thrilled to do this competitive infanticide strategy? Any guesses? Yeah, part two, individual selection, kin selection. Kin selection, you don't see the competitive infanticide if the males replacing are close relatives, by exactly that logic. So then you begin to see some more elegant stuff going on. In some cases, in a bunch of species, instead of males killing infants, what happens is the presence of a new male causes females to miscarry, pregnant females to miscarry. One version of this, and this is seen in wild horses, the new male in there harasses pregnant females to the point of them miscarrying. Same exact logic again. In a whole bunch of rodent species, you see something far more elegant. The new male shows up, and if you are a pregnant female, the smell of a new male causes you to miscarry a litter. And the biology of that is completely worked out, going from the olfactory signaling, and we're going to hear tons about olfactory signaling in weeks to come, going from the olfactory system to release of a stress hormone that disrupts uterine maturation, and you abort. Olfaction-induced abortion. What's up with the female with that? Why is that a strategy for her to leave as many copies of her own genes in the next generation? If this guy is around, she's got a choice at that point. A choice, again, metaphorically, she has a choice at that point. Either she can go through the rest of the metabolic costs of pregnancy, give birth to these kids, and then they get killed, or at least she can take the lesser of two evils. She spontaneously aborts at that point, and soon after she is ovulating and has a chance again to pass on copies of her own genes. And this is the whole world of you put a male hamster in with a female who's just had babies and he goes and eats the kids. Oh, it's psychopathological. It's not psychopathological. Male hamsters are laboratory. If there are kids there, they're not likely to have been mine. That's the logical thing. Females having been selected for, if there's the smell of a new male, miscarry. And the same logic again is the smell of a new male unless he's a close relative of the previous one. You see the exact same thing playing out there. Now, all of this, of course, is very sort of male biased. In terms of this, the female is making the best of the situation. Female Langer, when some new male has come in and is intent on being infanticidal, and you've read evolutionary biology also. What do you want to do to maximize copies of your own genes in the next generation? Well, you've got this kid here who you're trying to protect. Would you protect that child to the point of your being killed? Probably not. And you don't see females doing that. Under what circumstance would a female be most motivated to defend her child to the point of her being seriously injured? Older females. Older females who are less likely to have another child after. You're on the scene there, and who else from day one would be most willing to get injured in order to protect the kid? The maternal grandmother, who has no reproductive potential at that point. You see, in these Langer movies, females will defend, not to the point of serious injury. The older the mother is, the more strenuously she defends, and the grandmothers defend even more. Following the same logic here playing out in that way. But you also have another option, which is to do something much more clever. And this is not in the world of the world of when you are pregnant. Because even in these species that are competitive infanticide, the new male is also perfectly happy to have a pregnant female. What have females evolved in a number of primate species? One of the great all-time ways of manipulating males. So you're this pregnant Langer monkey, and in comes this new male, and you know you're going to go through like 6 more weeks of pregnancy, and you're going to be enormously costly, and the damn guys, they kill your kids as soon as you're born, and what can you do, and all of that, and you come up with something very clever physiologically. You go into what is called pseudo-estrus. Estrus being the term for when female primates are ovulating or sexually receptive, are in heat, and most primate species give external signs of when they're ovulating. There's engorgement of all sorts of tissues around the vagina and rear end and stuff, and that humans don't do, but they have some of the physiological residues of water retention, for example. So you've got all these external signs of, I am ovulating, except you're not ovulating because you're pregnant. You are pseudo-ovulating. You are going through a pseudo-estrus. You generate the picture of it. So what happens at that point with this new guy who's just shown up, and here's this like 4-month pregnant female who nonetheless looks like she's going through estrus? Well, these guys sit there then and say, well, that's kind of nice, and they go and mate with her, and like 2.5 weeks later she gives birth, and you know what the guy's going to do. He's going to sit there and say, what's the gestation period of my species? That's it. what's it, like 5 months or something? I've been here 3 weeks and already she's giving birth. Whoa, what a guy, what a guy. And they fall for it. They fall for it every time. Females, by having these pseudoestrus things, the males are not infanticidal afterward when the female gives birth. So this whole world that initially making no sense at all. Oh my God, animals killing members of their own species, even babies, making vicious logic and implying issues of individual selection and the qualifications by way of kin selection. Next example, another one with a similar flavor. And yeah. Yep, and I think probably the most technical Darwinian way of explaining that is the well-known propensity of blood flow to either go to male brains or penises. They just get impulsive at that point. They don't stop to think and get the gynecology textbook that, there no doubt is an advantage for the tiny subset of male langur monkeys who can actually be prudent at that point and think through the logic. It works with males. They fall for it, suggesting that a lot of what's going on there is not a conscious cognitive strategy. And this is sort of not a facetious answer when by the end of next week we get to looking at how do animals recognize individuals. With a lot of species, there's not a cognitive strategy. You're not able to sit there and figure out, wait a second, this isn't making any sense. So.
The dragon sword. So your father is still in the sacred wilderness.
I think
he has entrusted me with the
I
sword
think
bond.
what it is that there's not a whole lot of males there who could make sense or who have been selected for making sense of the fact that mating has something to do with babies down at the other end of things. I think there's not been much selection for being able to cognitively do that or to have some sort of rule. If I mate with this individual, anytime she produces a baby, I'm going to be unaggressive to the baby unless it's within this sort of time span. I think that's a little too much to have asked to evolve in sort of a primate system. The males fall for it.
It is a shame.
Another version with a lot of the same logic. And this became clear, studies of baboons, savanna baboons in the early 60s, which is you would have some male, who male is having a tension with another adult male, and they are about to have some sort of clear fight, and one guy is obviously low-ranking, and he's going to get trounced, and here comes the big high-ranking male, and this guy is terrified, and what does he do? He looks around frantically and grabs a baby and holds it to his chest. 1950s National Geographic special. Why is he doing that? Because infants are comforting, because babies are cute, and everybody becomes less aggressive when there's a baby around. Because, oh my God, who would attack somebody holding an infant? What if the infant's injured? Okay, so that's the old version. So applying some contemporary interpretations, what do you wind up seeing? Along comes the threatening male. The subordinate male grabs an infant and holds it. And what you only begin to see after a while of studying individuals is it's not random which infant he grabs. And you know exactly what's going to come next. A very similar theme to the competitive infanticide. When males are doing kidnapping like this, and that is the term used, they're not randomly grabbing kids, they're grabbing kids who are likely to be the offspring of that male. And it is very clear at that point, mess with me and your kid's going to get it. And it's clearly not being played out in any sort of conscious level like that, but what you see is it's not random. And it comes with the sort of qualifiers that show you even more how the system is working. So you've got some guy who's a big, high-ranking male and has obviously fathered all sorts of kids in this troop, and you're about to trounce this smaller guy who grabs one of the kids who's more likely to be his than yours. Logical competitive strategy of essentially blackmail. You've got a big, high-ranking male who's dominating everybody else, except he only joined the troop 2 weeks ago. In other words, there's not been enough time for him to have kids yet. When he is threatening lower-ranking guys, they're less likely to kidnap against him than kidnap against a high-ranking male with a long residency. And what you find even more interesting, which throws you into this world of, well, are these guys consciously thinking about this, or do they just smell that that guy's kid, and do they just know? In primates, they're thinking about it. And the way to realize that is when they screw up and make a mistake. And this one, sort of years ago, I was watching some baboons, and there was this low-ranking guy. He was actually middle-ranking, and this high-ranking guy was coming at him, and he was a middle-ranking guy who critically was on his way down. He used to be a high-ranking guy. He was aging. In other words, he used to be reproductively active. In other words, there's some of his kids around in the troop. So he's sitting there, and here comes this terrifying high-ranking male who's coming at him, and our guy gets all agitated and nervous and looks around, and he grabs a kid, and it's likely to be his kid. He's grabbed his own kid. Oh my God, you're sitting there saying, I'm never gonna get a damn thesis out of this. These animals just ruined all the theoretical models. He's grabbing the wrong kid. So he's holding the kid there, and you're sitting there, and you could see the guy's about 5 steps away, and so he does that, and tosses the kid clear just as he's attacked. You will see they act as if they have made a mistake. They are consciously working around something like this. So this whole notion of coercive blackmail and kidnapping, again, it only makes sense once you begin to see these structures of individual selection, kin selection, reciprocal altruism. Another example, and this one makes perfect sense instantly. You look at the world of female primates and lots of different species, and there's ranking systems, there's a hierarchy, and what's the hierarchy built around? You get a rank one below that of your mother. You inherit your rank. Your mom is the alpha female, you're her first daughter, so you're number 2. And as soon as she has another daughter, your kid sister is number 3. Until you have a first daughter who pushes your kid sister down one step. In other words, the dominant hierarchies amongst the females are entirely nepotistic. Whoa, how can you explain that? Part number 2, kin selection, dominant systems in all sorts of social species are built around the nepotism of relatedness. So that makes sense. Next one, next interesting notion here, which is one of those choices that are not conscious choices, but you're about to get pregnant and you have a choice as whatever species you are. Do you want to have a male or a female? Or do you want to have a litter of males or females? Or do you want to have a litter that's predominant male or predominantly female? And it comes down to an issue now of 2 things, which is how much does it cost to have a female versus a male during pregnancy? And what are the reproductive probabilities of having a male versus a female? Back to this issue, you are in a big tournament species, and as we saw, there's high degrees of male variability in reproductive success. 5% of the guys are accounting for 95% of the matings. So you sit there, and what you've got is a rule that if you go for a son, like 90% of sons running around there are never going to reproduce, and you hit the jackpot and you have the 5% at the very top, and each of them are going to father 30 different kids. Going for males in a tournament species is a big gamble. It's a risky move. Going for a female, however, there's no female primate out there who has 420 kids because she lays eggs like a salmon. Most females there, unless they have some fertility problem, they all have something roughly 1 to 5 kids or so over the course of the life of an old world primate. So female variability is way down. In other words, what's a conservative strategy to pass on copies of your genes? Have a daughter. What's a riskier strategy? Have a son. And what that immediately predicts is 2 things. Number 1, you look in dominance hierarchies, and the prediction is that females who are high-ranking should show more of a tendency towards having sons than daughters, and females who are low-ranking, exactly the opposite. And that's what you see in a bunch of primate species that have this sort of structure. Next prediction. You should then predict that when ecological circumstances get tough, when times are tough, you want to go for the offspring that costs less. A female fetus is less calorically demanding than a male fetus. Male fetuses cost more to bring to term than females do. The prediction should be during times of ecological pressure, the percentage of females being born should increase. And as a measure of the fact that males are more expensive as fetuses, something like 53% of fertilization in humans are males. About a 53 to 47% ratio. And over the course of pregnancy, the increased metabolic vulnerability of male fetuses are such that by the time birth comes around, it's around 51-49. And it's not until adolescence that it flips over to the typical pattern of female dominance. You've got to have more male fetuses to start off with because they are more expensive, more vulnerable. So this prediction during times of ecological duress, you should get a bias towards more females being born. The 50-50 ratio is skewing in that direction. And that's precisely what you wind up seeing. And you see all sorts of examples of this in humans, for example, during periods of famine, food deprivation, the ratio of births skewed towards females. What you also see as a measure of that is among humans, a boy giving birth to a boy statistically decreases the body weight, is likely to decrease the body weight of the next offspring. It's expensive having one of those males. And what you wind up getting then is fluctuation as a function of your dominance rank. If you're high ranking, it's almost always worth the gamble to go for one of those high risk, high payoff boys. If you're low ranking, go for the far more conservative female. You've got this fluctuation around this 50-50 ratio. And this was something worked out by one of these sort of founding figures of sort of modern evolutionary thinking, a guy named Robert Trivers in the 1970s, sex ratio fluctuation as a function of social context. And people have gone and looked, and it's precisely this. You get an interesting bit of conservatism in this, though, which is there's some circumstance where it makes perfect sense for you to have a gazillion daughters because that's the time to do it. And at some point, you're having a gazillion daughters and everybody else is having a gazillion daughters. And suddenly, males become really valuable because there's not a whole lot of them around. So the logical thing to do then is to switch over and start having males. And everything else being equal, after a while, with a predominance of males, it's going to make sense to switch over to females. You have density-dependent selection. You will always have oscillating around 50%. Whichever sex is in the smaller number, that one is immediately more preferable. Poor ecological conditions pushes you this way, dominance rank pushes you one way or the other, but in any of those circumstances you have an oscillation around the mean. Whichever is more common is less desirable. So sex ratio fluctuation. More stuff. Role of kinship, for example, in vervet monkeys, we talked about that the other day. Play the sound an infant alarm call, and the mother gets all agitated, and everyone else looks at the mother. They know that she's the mother, sort of demonstration of awareness of kinship there. Now, another very interesting social structure to some species, which is we've seen all of these tournament species. You have one high-ranking male mating with lots of females and all that sort of stuff in polygamous systems. What about polyandry? Circumstances where a female is mating with multiple males. What about circumstances of stable polyandry where you get, in effect, the inverse of a harem? You get a single breeding female with a number of males. And what is seen with a very, very high predominance is when polyandry occurs, you get a type which is called adelphic polyandry. And the second I tell what it is, it will make perfect, wonderful sense. Okay, two male lions sharing a pride, that's like not what you're supposed to see. It's one male lion and Mufasa and his brother gets pushed out of there, and that's not what you see when you study Disney lions. There's supposed to be only one male as the breeder and the pride. And occasionally you see these prides where instead there are two males. How can they pull that off? They should be doing competitive infanticide, all of that. Who are the two males? Yes, you guessed it, somewhere up there before, it'll be two brothers. When you see cases of two male lions sharing a pride, or flip the other way, when you see a pride being willing to tolerate two males in there instead of one, very, very high likelihood that they are brothers. This technical term, adelphic polyandry. And you wind up seeing one totally wild example of this in humans. As I went over the other day, looking at our humans tournament species, are they pair bonding? We're somewhere stuck in between. We're terribly confused. What you see is most cultures traditionally allow polygamy, but most people are not actually polygamous, all of that. And somewhere in there, you got to ask the same question, hey, is there any polyandry going on with humans? And there is one wild cultural example of this, and this is seen in traditional Tibetan society. And there you get Adelphic polyandry. You have the following structure in rural areas. A woman will marry a man, and in the process, she will marry him along with all of his brothers. All of his brothers, she marries the entire lot of them. It is Adelphic polyandry. And you see that, and like it's down to the point where here's this woman with her husband and his younger brother, and his younger brother all the way down to this infant that she's holding, who's the youngest brother, who's now one of her husbands. And that's what you see. What's the explanation for this? This is a pattern that you get in agriculturally very impoverished areas where you got a problem. You've got 5 sons or whatever, and with a pattern of land inheritance where you would otherwise split up the land amongst the 5 sons, that's going to put each of them below subsistence level. What you need to do is have a way in which they remain as one reproductive unit. So you don't split up the land. You see this Adelphic polyandry, a woman marrying this whole bunch of brothers, and it's in circumstances where it's trying to keep the small farm plots from being broken up. But again, the logic there is the same as you see in lions with 2 brothers sharing a pride. What else? Now, somewhere in there, you begin to get in a realization that not only do you have males competing with males for reproductive success and females with females, but there's intersexual competition. How could this be? How could a planet that comes up with Valentine's Day have intersexual competition? What is obvious is the reproductive interests of any given individual may not be exactly the same as the individual she or he mates with. What would be an example of this? Here you have a species in which males are migratory. It's a tournament species. A male shows up during a mating season, lots of aggression, lots of secondary sexual whatever, and he mates with a female. And she has kids. Hooray, they have parented a offspring and they've shared their genes and increased their reproductive success, except this being the species, this guy is going to pack up and leave the next day. In other words, he has no investment in the future reproductive health of that female, whereas she sure does. She would be delighted for this offspring to thrive and prosper, but perhaps not at the cost of her future reproduction. What we see here is between the male and the female, different reproductive strategies. And this opens up a whole bizarre world of intersexual competition. Here's one really bizarre manifestation of this. Okay, basic Mendelian genetics. Those of you who got the catch-up this week should have a vague sense of this by now. Hardy-Weinberg ratios. You've got one parent has one genotype just to distinguish the parents and changes the colors there, all of that. What is it that isn't bothered with from day one when you learn these sorts of things? It doesn't matter which one is the male and which is the female, which is the father and which is the mother. This is just one of them contributes this profile, one that, what pops out the other side. Basic Mendelian genetics, when making sense of pedigrees, it doesn't matter which parent is contributing the homozygotic profile, the heterozygotic, it doesn't matter which parent. But then there's a whole world of genetic traits where in fact it does matter which parent it comes from, because the same combination of alleles will function differently whether it came from the mother or from the father. And this was a field that sort of emerged in the early 1990s, and these are called imprinted genes. Imprinted genes, these are genes where, for our purposes to define them, these are genes which have different manifestations, different phenotypic consequences. These are genes that work differently depending on which parent they came from. Mendel rolling in his grave, this is not how it's supposed to work. This obscure world of imprinted genes violates this. They work differently depending on which parent you get it from. dramatic foreshadowing. When we get to the ethology lecture, the word imprinting is going to come up again in a totally different sense. This is a purely molecular term right now. Okay, so you get imprinted genes. Imprinted genes, people began to figure this out in the 90s that, wait a second, we've got a completely different profile in these individuals with a mutation in a particular disease. Here we have these two very different diseases, and they turn out to have the exact same mutation in the same gene. What's up with that? If you get that gene from this parent, you get this disease. If you get it from that parent, you get that disease. Totally boggling, making no sense at all. First thing to have to be solved. What's a mechanism for actually making a gene work differently depending on which parent it comes from? For those who care about these sorts of details, it's a biochemical process called methylation. You methylate the gene in one parent's genome and not the other. Don't worry about the details. The main way is there is a genetic mechanism for making genes work differently depending on which parent it comes from. So, okay, we know how to do it. Why should this happen? Why does this make any sense? And it took a while, and people began to see patterns to this. And finally, this was put together by an evolutionary biologist at Harvard named David Haig in the early 90s, who came up with a unifying theory which explains it all. And it's very cool. Here's what you see. You begin to look at imprinted genes. And for our purposes, the way we can describe them as, this is a gene where if you get it from one parent, it does what it's supposed to do. If you get it from another parent, it's silenced. It never works. It's methylated into silence. These are imprinted genes. So he began to notice something. When you look at imprinted genes where they are active if they come from the father, they all tend to be genes that promote fetal growth. They're all genes that increase fetal metabolism. They're all genes that make for a thicker uterus. They're all genes driven by hormones coming out of the fetus. They're all genes that push for greater fetal development. And then you look at the imprinted genes coming from the female, and they're all genes that tend to slow down fetal development. What have we got here? We've got that intersexual competition played out precisely there. You've got this male hamster who's going to mate and never be seen again because he's in this category. And what does he want? He could care less what happens to the future reproductive success of this female. He wants this offspring of his to survive. The male imprinted genes push for greater amounts of fetal growth. And the female one's saying, well, that would be great if this kid survives, but I also have a future reproductive life to think about. Her imprinted genes tend to counter it. What would this look like? One of the imprinted genes, first one's identified, which comes from active from the male, codes for a protein called insulin-like growth factor. Who cares what insulin-like growth factor does? What's clear from the name is it's a growth factor. It's one of those things that make fetuses grow more. It promotes fetal growth. And then meanwhile, the female hamster has a cognate, an opposing imprinted gene. What's that one for? That codes the gene for the insulin-like growth factor receptor. And what her version does is make for a less responsive receptor. The male is pushing for more of the insulin-like growth factor. She gets a receptor that's not as responsive, and you literally have this co-evolutionary arms race there of male imprinted genes evolving to push for more and more fetal growth, female ones pushing for less and less fetal growth. And you see that coming out most apparently when you see mutations in some of these genes. For example, another pair of imprinted genes, one from the father, one from the mother, and what you see there is the mother's version. The father's version promotes invasion, placental invasion into the uterus, and that's actually the term that gynecologists use, invasion of the fetus into the uterine wall. It promotes more of that. The female version slows it down. So what if you have a disease where the male gene relevant to this is mutated and knocked out of action? You solely have the female input, which is decreasing fetal implantation into the uterus. What have you got then? You've got a disease where fertilized eggs don't implant. On the other side, suppose there's a mutation in the female part of this pairing. And as a result, all you have is the male input driving for more and more aggressive fetal invasion. What do you have? You wind up with one of the all-time bad cancers you don't want to get, choriocarcinoma, a cancer of the uterus, because it's growing completely out of control, being prompted by the fetus. When you take out each of the voices through a mutation, you see that normally you're having this tilting, this balancing of competition between males pushing for more fetal growth at the expense of the future reproductive success of the female, females trying to slow it down. All of the imprinted genes show this. Amazingly, people now have even identified imprinted genes that don't work until after birth, and they work in the brain. And what do? The male-derived one does things like make for infants that suckle more. Ooh, get more calories out of mom. And the fetal version tends to blunt some of the suckling reflexes. It's all built around the strategy of males and females do not necessarily have exactly overlapping reproductive strategies. So another version of it is a male gene, which is pushing towards expression of a fetal enzyme, fetal lactogen, placental lactogen. And what that does is it makes it easier for the fetus to grab sugar out of the bloodstream from the mother. And the mother tries to counter it, and if she's not very effective at doing it, or if she's rather over-effective, you get pregnancy hypoglycemia, pregnancy diabetes. Suddenly, this is mom having a fight with her offspring over how much calories they're going to get, the offspring being driven by an imprinted gene from the father. Totally cool, totally interesting. There should be a problem lurking here, though, which is this is a pattern you only see in tournament species. You got a pair bonding species, and there's no reason why a male should be saying something like, ooh, I want to ruin her future reproductive success at the cost of her giving birth on a pair bonding vole, and I want her to give birth to a child the size of an elephant who's going to survive, and who cares about her future life? They're pair bonding. They're in it together for the rest of time. You don't find imprinted genes in pair bonding species. You find imprinted genes in tournament species. And thus we get back to that same issue the other day, looking at tournament versus pair. Where do humans fall into this? And what we've already seen is if humans can come up with choriocarcinomas and things like that, we have imprinted genes. Same punchline again as the other day in terms of the number of genes we have puts us somewhere in between tournament and pair bonding species. Again, we are terribly confused. Okay, let's take a 5-minute break and we will pick up with more examples. Okay, let's get going again. Two good questions just now. One is, where does homosexuality fit into all of this? And where it fits in is about 30 minutes worth of the sexual behavior lecture sometime in mid-May. It is a challenge for some of this thinking. The second good question was, am I capable of speaking louder? I will try. I mumble. Okay, pushing on. So we've just brought in this whole bizarre, unexpected world of trashing the Hallmark cards of intersexual competition. Another example of it, a really interesting one. This was work done by a guy named William Rice at Santa Cruz over some years, looking at a number of different fly species. Female flies are polyandrous. They mate with a lot of different males. And you're a male who've just mated with a female, and what you would like more than anything in order to pass on copies of your own genes is not have some other guy impregnate her, some other male fly guy who's intimidating you, not have some other guy do it. And suddenly what you see is this interesting world where, because of the mating frequency of the females, she will have sperm from a number of different males inside her at the same time. And suddenly we get this very strange world, a whole field of research of sperm competition. Sperm competition of relay teams and gold medals and all of that, of sperm competing with the sperm from other males. And what you see is, in fly species, the sperm of males make toxins that kill the sperm of other males. Whoa! That's very elegant. That's very elegant because it in part requires you to come up with a toxin that isn't toxic to itself. You could solve that, all sorts of molecular tricks for doing that, but this makes wonderful sense. You increase the likelihood of you, the sperm, reaching the goal line and killing the other guys and all of that, except there is a problem for the females, which is these toxins that the male sperm release aren't such a hot deal for the female's health. And what Rice did were these really interesting studies. I won't go into the details of it in large part because I still don't understand what the guy did, but he was able to somehow take populations of male and female flies, and he would hold the females so that they could not evolve in response to whatever was going on to the males, male-male competition for reproduction, while he held constant female-female competition. The females were not evolving, the males were. And what he saw over the course of 30 generations was this male-male sperm competition was such that the male sperm were making such powerful toxins that they were shortening the life expectancies of the females. Whoa, that's not smart. Whoa, that makes perfect sense. Houseflies are not your pair-bonding swans dying in each other's arms for life sort of organisms. It's a classic case of the male only has an investment in the current reproductive bout, and if he can wipe out the competitor's sperm, and oh bummer, the female has now got all sorts of rotty necrotic lesions in her vagina. This is not a very reproductively effective female fly down the line. What do I care? I'm off to the next pile of cow dung after that. You have that same structure there, but this is a bummer for the females. The males left to their own competition will evolve more and more toxic sperm, which exacts more and more of a price on the future reproductive success of the females. Now, Rice flips things, and now he takes these populations and he holds the male evolution constant and allows the females to evolve, and over the course of 30 generations, they have evolved means of detoxifying the sperm. So here we have male-male competition inadvertently being played out in an intersexual realm as well, where again, this co-evolutionary arms race, totally bizarre, sperm killing each other and sperm damaging the female, and that makes no sense if you do for the good of the species. Again, very logical in the context of these models of evolution. Okay, final example. And here's one that will become very relevant way down the line when we're looking at things like the biology of aggression. So you've got these social primates, and in lots of social mammals, you've got this pattern, which is one of the genders picks up around puberty and moves to another group. It's to avoid inbreeding and everybody having 6 fingers and tails if you're a species that doesn't have a tail, and it's perfectly logical, and what you need is just some sort of pattern is that the females or the males of the species which disperse. And no one among the primates has ever found a good rule for which primate species have female exogamy or male exogamy, which gender leaves at puberty, but there's variability. In chimps, it's the females who leave at puberty. In gorillas, it's the females who leave at puberty. In baboons, macaques, a few other old world monkeys, it's the males who leave at puberty, with a very critical implication. So you've got baboons. Baboons, you've got a troop, and you look at the adult males and adult females in there. The adult females grew up in that group. The adult males grew up someplace else and emigrated into here at puberty. In other words, among the adult females in this group, they're all relatives. Among the males, there's no relatives. Meanwhile, next door with the chimps, it's the females who have left at puberty. You look at the mature animals in a group, and it's the males who are there with all the relatives who have been there all their life, and the females who have no relatives. So, asking a question, comparing baboons and chimps, which of those species has higher levels of male-male aggression within the group? Baboons, yeah, because they're all brothers at arms in the chimp group there, and none of the adult males in baboon troops are typically related to each other. Very high levels of intermale aggression intra-group. So which species on a certain level has put us to shame when it comes to the invention of warfare, of fighting between groups? Chimps. because you've got bunches of males who cooperate, because they are relatives. And what you wind up seeing with chimps, and we will eventually get to this, is you see things that are now termed border patrols, where a bunch of males from the group will patrol the edge of their territory. If they encounter a male from the group over there, they will attack, they will kill him. As documented by Jane Goodall, taken to an extreme, the males of one group will eradicate another group. And if the rule is, I am killing this guy, we are killing this guy, not because we don't like the look on his face, but because he is a member of that group, this is the United Nations definition of genocide. Chimps have not only come up with organized warfare between groups, they have come up with a chimp version of genocide. What's the driving force on that? One of the truly scary things on this planet, which is when all the males living next door are getting along with each other. Because when they do, they suddenly start looking over at this side. Organized males driven by female exogamy in chimps is where the warfare pattern comes from. And as you can guess, when we get to thinking about the biology of aggression with humans, we are suddenly in this world of taking non-relatives who are military recruits and convincing them to pseudo-kinship themselves into being a band of brothers, increasing the sense of kinship there. Chimps do this all on their own. So now making sense of which primate species have high levels of male-male aggression within group versus between group are ones where it's the females who leave at puberty. All these guys have been together since they were terrorizing other kids in kindergarten. They're an organized group against each other. You get intergroup organized conflict in primates that have female exogamy, females leaving at puberty. Okay, so we've now worked our way through a bunch of examples here and how to apply individual selection, kin selection, that sort of thing. A fourth branch has come into the field in the last decade or so. And this is one that has had a lot of controversy, in part because what it is built around is mistaken by a lot of folks as being an outdated concept, but in part because what it is actually implying is very controversial in some realms. And this is this business of group selection somehow sneaking back in the back door there and becoming relevant to the evolution of social behavior. Group selection, you remember Wednesday, all the wildebeest there, and the elderly wildebeest pushes his way forward and distributes his bank account and then leaps into the river and the crocs and all of that, and that makes no sense at all, and because animals don't behave for the good of the species and individual selection, he got pushed in. Nonetheless, a notion of group selection has come back into thinking, but it occurs only under a couple of specialized circumstances. The version that we just trashed, which was the version okay, how many of you guys know who Marlin Perkins was? It's come to that. Marlin Perkins, how many of you guys know what, does Mutual of Omaha, does that still exist? Mutual of Omaha's Wild Kingdom? Is that still a television program? Yes, no, when you were young? Did that exist? Okay, is it still sponsored by Mutual of Omaha? They still do. That's great. Okay, because that one has been going for decades and decades, and it was always, they somehow always have to segue the Mutual of Omaha in there during the commercials, building it just as rhinos will mate for hours on end, you want fire insurance for your home or some such thing. But in its original couple of decades, it was hosted by a guy named Marlin Perkins, and Marlin Perkins taught most of America their evolutionary biology. Marlin Perkins is the guy who taught everybody behaving for the good of the species and that sort of thing. The first version of group selection that got trashed in the 60s was Marlin Perkins group selection. The group selection that's sneaking back into the field is very different. Two different versions. First one, something you will get in various populations. You've got a population of something or others, and some biogeographic event occurs which causes a subset of them to get isolated from everybody else. A land bridge disappears, or somebody drops a lake there, or who knows what, but something isolates a small subset of the population. So these guys go about reproducing by their standards, and these guys go about reproducing by their standards. And what's going to be a characteristic very soon of the smaller population, they're going to be more inbred than this population, simply because they're smaller. They're going to have a higher degree of relatedness among individuals. So now we throw our second piece of our 3 building blocks, throw in kin selection, and what that immediately predicts is levels of cooperation will be higher in this group than in this group, because the higher degree of relatedness. That's great. And that's great because once we see how sort of cooperative systems have a larger payoff, you will get a crystallization so that everybody, the second or third cousins, are eventually going to have to be just as cooperative as the siblings are with each other. You are going to fix a trait of cooperation in that population at a high rate. So notice here, in this case, we have a high degree of cooperation driven by kin selection, whereas these guys are going about their usual savage at each other's throats business. Biogeographic event reverses, and these guys get reincorporated into the main population, and they're so different by then that they get a different color. And what you've got then is, here in the large population, here is a nucleus of animals who are cooperating. And here is the huge unwashed mass of the ones who are not. And everything we know about reciprocal altruism, all of that means that these guys are going to start outcompeting these guys. And the model that is used is cooperation will have to crystallize outward. Animals will have to join in these cooperative patterns because these guys will outcompete them. What do we have? This is called a founder effect. This is a small population that, thanks to being inbred, has fixated some trait that's advantageous, where the evolution here moves faster than in here because of the smaller population with the inbreeding. Some adaptive trait comes in there, they get reinduced into the main population, and this founder-driven trait quickly spreads throughout the population. And what you get here is, if the same thing is occurring in the realm of behavior, reciprocal cooperative behavior, somewhere in here you have a transition from this being a kin selection phenomenon to this being a reciprocal altruism phenomenon. So that stands as one of the models out there for how do you jumpstart cooperation in non-relative populations. You use inbred founder populations to drive it there. And just to have a metaphor, all you need to do, think about of specialized circumstances. The group selection that's sneaking about reproducing by the geographic Marlin Perkins, and Marlin Perkins taught most of America their evolutionary biology. Marlin Perkins is the guy who taught everybody behaving for the good of the species and that sort of thing. The first version of group selection that got trashed in the 60s was Marlin Perkins group selection. The group selection that's sneaking back into the field is very different. 2 different versions. First one, something you will get in various populations. You've got a population of something or others, and some biogeographic event occurs which causes a subset of them to get isolated from everybody else. A land bridge disappears, or somebody drops a lake there, or who knows what, but something isolates a small subset of the population. So these guys go about reproducing by their standards, and these guys go about reproducing by their standards. And what's going to be a characteristic very soon of the smaller population, they're going to be more inbred than this population, simply because they're smaller. They're going to have a higher degree of relatedness among individuals. So now we throw our second piece of our 3 building blocks, throw in kin selection, and what that immediately predicts is levels of cooperation will be higher in this group than in this group, because the higher degree of relatedness. That's great. And that's great because once we see how sort of cooperative systems have a larger payoff, you will get a crystallization so that everybody, the second or third cousins, are eventually going to have to be just as cooperative as the siblings are with each other. You are going to fix a trait of cooperation in that population at a high rate. So notice here, in this case, we have a high degree of cooperation driven by kin selection, whereas these guys are going about their usual savage at each other's throats business. Biogeographic event reverses, and these guys get reincorporated into the main population, and they're so different by then that they get a different color. And what you've got then is, here in the large population, here is a nucleus of animals who are cooperating. And here is the huge unwashed mass of the ones who are not. And everything we know about reciprocal altruism, all of that means that these guys are going to start outcompeting these guys. And the model that is used is cooperation will have to crystallize outward. Animals will have to join in these cooperative patterns because these guys will outcompete them. What do we have? This is called a founder effect. This is a small population that, thanks to being inbred, has fixated some trait that's advantageous, where the evolution here moves faster than in here because of the smaller population with the inbreeding. Some adaptive trait comes. Cooperation will have to crystallize outward. Animals business. Main population, and they're so different by then that they get a different color. And what you've got then is, here in the large population, here is a nucleus of animals who are cooperating. And here is the huge unwashed mass of the ones who are not. And everything we know about reciprocal altruism, all of that means that these guys are going to start outcompeting these guys. And the model that is used is cooperation will have to crystallize outward. Animals will have to join in these cooperative patterns because these guys will outcompete them. What do we have? This is called a founder effect. This is a small population that, thanks to being inbred, has fixated some trait that's advantageous, where the evolution here moves faster than in here because of the smaller population with the inbreeding. Some adaptive trait comes in there, they get reinduced into the main population, and this founder-driven trait quickly spreads throughout the population. And what you get here is, if the same thing is occurring in the realm of behavior, reciprocal cooperative behavior, somewhere in here you have a transition from this being a kin selection phenomenon to this being a reciprocal altruism phenomenon. So that stands as one of the models out there for how do you jumpstart cooperation in non-relative populations. You use inbred founder populations to drive it there. And just to have a metaphor, all you need to do, think about in some city, there's some occupation, some mercantile, something or other, that there's a gazillion of them and all sorts of people work in that. And there's a subgroup of people there who work in this who are all related. And as a result, they do something cooperative and kin selective. They make low interest loans to each other. And as a result, they're more successful at business. And they get incorporated into here. And what you then have is this economic force that everybody else has to join in to this cooperative business of trusting each other, or low interest loans, or I trust you like a sibling, you don't have to pay me for next week. Used to be that only siblings trusted each other like siblings, but a force there for more cooperation, a driving force of inbred kin selection initially. So that's one way in which people are thinking about group selection, and here you now have this group outcompeting this because of this trait, and people actually use terms like crystallization. The trait of cooperation will crystallize outward and fix in the whole population. So that's one version of it. And this is one, again, we will come back to. This is one of the ways to jumpstart cooperation in a world in which there is no reciprocal altruism. Because you see the problem with that. We all have learned tit for tat is the best one, except forgiving tit for tat might be better, except Pavlov might even be better. And all of those are built around this one requirement, though, which is somebody has to make the first move of being cooperative. That's got to jumpstart the system. And what we all know is, in a system that doesn't go along with the rules of can't we all sing Kumbaya, what happens is the first one in the first round who does something cooperative is some schmuck who's one step behind everybody else for the rest of time. There's no way to get these cooperative systems to evolve initially. We'll see there are ways, and this is one of those. Have it driven initially by kin selection, and then throw it into the general population. So that's one version of where group selection is crept back in. Another version is sort of a more generalized form of it. And it could be illustrated, okay, the following example. You've got chickens. You've got chickens with an array of traits, and you can see there's two types of chickens. One chicken is super aggressive and beats on everybody else, and she lays lots of eggs. Another chicken, far more pacific and introspective and less aggressive and has fewer eggs. So now you take any one of the high aggression females and one of the low aggression guys and put them together, and who's going to leave more copies of her genes? The high aggression female laying lots of eggs. Now instead, you have a whole group of the high aggression females and a whole group of the low aggression females. What's going to happen in the high aggression females? They're all so aggressive that they all injure each other. They all stress each other into lower fertility rates, all of that. And suddenly you have this very important world in which A can dominate B, but where groups of B dominate groups of A. And it's in that realm that you suddenly get a push towards group selection. When selection on that level, a world of any individual being out-competed because of a trait, but as a collective, and bringing in that word, not randomly, as a collective and the collectivist future of low-ranking chickens to throw off their chains. Once you have a group of them, the same traits that could be disadvantageous on an individual dyadic level as a group outcompetes the others. And when you do that, you've suddenly got group selection in a classical form where these animals, because of their traits, they are not behaving for the good of the group, but the very traits that are disadvantageous individually are advantageous as a group. And it's another way of thinking about this. If you indiscriminately make low-interest loans to everybody on Earth, you are very readily falling into this category. If you are part of a group that makes low-interest loans to each other, suddenly you outcompete the other. Fascinating book, evolutionary biologist named David Sloan Wilson, who's been the main person pushing for this idea for decades, has a very broad range of intellectual interests, including religious history. And a number of years ago, he published a book called Darwin's Cathedral. And he analyzed the birth of clusters of new religions over time as examples of founder effects and group selection type properties, and he wound up in great detail analyzing Calvin and the starts of Calvinism, and Calvin and his sidekick Hobbes in Zurich in the 17th century, or whenever it was that Calvinism started, of analyzing how there were all sorts of these little religions popping up there in these little city-states, and what it was about Calvinism in, I think it was Zurich, that took off, where they established some of these inbred cooperative patterns and then beat the pants off of all the other city-states, and soon all of Central Europe was Calvinist. Really interesting book in that regard. Okay, so back has come this whole possibility of group selection. And out of that has come the more broad way of thinking about it now, which is multi-level selection. A very important concept here, which is sometimes selection occurs entirely on the level of a single gene. In other words, have a gene with bad enough of a mutation, and it doesn't matter how many great other genes you got going for you, selection will be decided entirely on the basis of one gene. As we saw in our principles from the other day, much more common selection at the level of the individual. An aggregation of the genotype playing out in terms of phenotype, all the different traits there. But what this introduces is some circumstances where selection is at the group level. And it is never at the level of how an individual's trait plays out individually. In a group, it's how this trait played out individually emerges as a group behavior that you get group selection. And this marks this great piece that's come between this David Sloan Wilson and the guy who's probably the most towering figure in this whole field, this guy E.O. Wilson, Edward O. Wilson at Harvard, who David Sloan Wilson and E.O. Wilson spent years not getting along with each other because E.O. Wilson is very strongly individual selectionist model guy, and thus he hated group selection. David Sloan Wilson the other way around. Each of them were endlessly invited to the wrong parties because of the same shared last name, and all sorts of social awkwardness there. In the last few years, I don't know what happened. I don't know if they arranged marriages between their grandchildren or something, but they arranged some sort of truce, and Wilson published a paper last year basically saying, whoa, you know what, some of the time I'm right and some of the time he's right, and isn't that great, and can't we all get along? And delineating circumstances where individual selection, kin selection, reciprocal altruism is going to dominate, circumstances where selection is going to get played out at the group level. They love each other now, they have formed a new language, they share burial customs, and this sort of constitutes a great resolution. Yeah, depending on the circumstances, the most important thing in evolution might be a single gene, a single organism, or a single group. And the power of a group and aggregate traits that work lousy in the individual is going to be really important down the line when thinking about aggression and stopping aggression and things of that sort. Okay, so all of this is great. This multi-level selection now showing sort of a fancier, more sophisticated way of thinking about individual, kin, reciprocal circumstances that work, circumstances where this is the most interesting thing. So that's great. So now we begin to look for a first pass at where humans fit into this. We've already seen, in terms of all of these traits, tournament versus pair bonding, we're somewhere in between. In terms of imprinted genes, we're somewhere in between. These are specific. How do we begin for a first pass to think about humans, human behavior fitting into these frameworks? Two problems or two issues that are all we'll touch on at this point. The first one is there's this temptation as soon as you think along these lines to discover, oh, isn't that interesting? Langur monkeys will have infanticide. Male langur monkeys will kill the kids of some other male. Human males do that sometimes. Oh, langur monkeys might be an interesting evolutionary model for where infanticidal behavior came from in humans. Oh, isn't this interesting? Dominance is passed along by males. We do that. Isn't it interesting? Kidnapping somehow occurs, and before you know it, you have a world in which people are saying, well, damselflies do something that look like rape. So, we've got a damselfly model of human behavior in that realm, and two things are happening there. One is taking the traits, a small number of traits in some other species, and inflating the relevance it has for understanding us. The other danger is somewhere creeping below the surface is, you see, it happens in other species. It's kind of natural. It's kind of inevitable. This is a whole world in which people have run off with how this applies to humans in a completely distorted way. We are unique as a species, as is every other species. We are unique by applying the same evolutionary rules that every other species has. That's the commonality, not the outcome. The other challenge is constantly going to be that you get all these great predictive models, and then humans go and screw it up by having quirks and idiosyncrasies and personalities and stuff like that. All I can say in that realm is back to Wednesday, if making sense of which lions are going to go to the scary speaker in the bushes there revolves in some part around personalities, humans, by the time you get to us, blindly assuming these models apply is going to be very sort of unshaky, very shaky. Okay, so just the first pass at how one will have to be cautious for the rest of the course in applying all these models. Now, far more critically, is looking at what are the criticisms that have been brought against this whole framework. This framework of social behavior has evolved to maximize number of copies of genes driven by kin, driven by reciprocal altruism. Group selection is a rare trait, but when it does, it follows these properties. This is how you explain all the behavior. Know these 4 or 5 rules, and not only can you look at 2 skulls of different sizes, but you can explain all of behavior and all of human behavior, and it's all biology. And eventually, as E.O. Wilson said in this landmark book of his in 1975, Sociobiology, eventually all of the social sciences will be under the wing of evolutionary biology. No surprise, the social scientists were not very pleased with that proclamation at that point, but this was this notion that this was going to explain everything. So what are the criticisms that have been leveled at this style of thinking? As we see, there are a lot of them. All of the stuff we've been thinking about, this basic notion of applying the Darwinian rules as to why giraffes have to have hearts of certain size to behavior, there's 3 features that are coming through throughout. The first one is the notion of heritability of traits. Because what we've been discussing throughout the last lecture or so is there's heritability of the tendency to kidnap infants at the right strategic time. There's heritability of the tendency to decide that you like males with big fancy plumage and long canines as opposed to, there's heritability to deciding that your daughter should be highest ranking in the true brother of that one's heritability. An assumption of heritable behavioral traits runs through every single one of these discussions. The next theme that runs throughout all of them is the notion of adaptiveness. If some trait has emerged out of the evolutionary murk, if some trait has been selected for, selection implies adaptiveness. The notion that everything you see has an adaptive explanation for why it's emerged evolutionarily. The third principle, which is not explicitly stated in this field very often, but is implicit to everything about it, is these processes of evolutionary change are gradual. At every generation, if by getting into some better behavioral strategy for playing prisoner's dilemma, you leave 1% more copies of your genes than everybody else, come back in 100 zillion generations, and that trait will have spread. All of this is played out generation after generation so that evolutionary change is occurring in gradual incremental steps. So what do you do with these three starting points? The first one, an awful lot of the next 2 weeks, we'll be looking at how other disciplines look at the issue of how do you figure out when a behavior is genetic. And one of the first things we'll learn is how that phrase means absolutely nothing whatsoever. Maybe a better way of stating it is, how do you figure out when a behavioral trait has a genetic influence? And as we'll see, even the safer way of saying it is, how do you figure out in which environment a certain behavior has a particular genetic influence? How do other disciplines go about saying, oh, it's genetic. It's genetic. In this field, among this sort of sociobiological thinking, what you do is an inverse style of proof. You say, okay, if a trait has a genetic component, you would expect it to be heritable along these lines. If it's more adaptive, it will increase its frequency, all of that. And look, that explains where competitive infanticide has come from. Show me a more explanatory model. Show me a model that is more predictive, and until you can show me one, I'm assuming this trait has some heritability. And we'll see how, by next week, the more molecular people rip this thing to shreds. Tell me, when you're talking about heritability of some behavior, show me the damn gene. Show me the sequence of DNA. Show me how that produces the behavior. This is where this field ends, saying, oh, we can inferentially sort of decide there's a genetic influence. As we'll see by next week, this one piece is the main thing attacked by a number of other fields. Next piece, the adaptiveness. The notion that everything that has evolved under the scalpel of evolutionary demand for optimization, all of that, and the critics of this view call this the adaptationist fallacy. The notion that everything is there for a good reason. And what they often do in terms of making fun of that is that, oh, you look at people who do this kind of evolutionary biology with this adaptationist emphasis, and it's one big just so story. Why do giraffes have hearts with thick muscles? And here's the story I make up. And if my story is better than yours, I get tenure and you don't. Why is it the zebra's got stripes? Why is it that males kill infants in the species or whatever? That everything is adaptive, and what you need to do now is come up with a story to explain why it's adaptive. And if it fits some of those principles of individual and group, etc., then you win. You've got the best just so story. And what is constantly brought as a sort of criticism of that is, show me experimental evidence that it is adaptive. By the time we get to looking at the field of ethology, you'll see a whole way in which people do that. Show me that it's possible to disprove it. Okay, tell me a just so story where, if that were the case, that would prove that this is not what's going on. You've got to have the rules of science. It can't just be you observe something and come up with the best story and you win. But what's most striking in sort of terms of criticizing this is some domains in which there are traits that have not evolved because they are adaptive. They've just evolved because they are excess baggage. And this has brought in a whole sort of criticism of the field. Now, some of the time when you begin to see, it's kind of hard to see where the adaptiveness is. It's because it's a more subtle feature of it. For example, you could look at squid. And someone who is enamored with fish and how cool and aerodynamic, aquadynamic fish are, and all of that will point out that squid are pretty lousy swimmers compared to all sorts of fish. They're pretty lousy swimmers compared to fish, but they're pretty great swimmers for something starting out evolutionarily as a mollusk. This adaptiveness has to be in the context of where it came from, where it got to. Okay, so that is a qualifier. Nonetheless, even factoring that in of hidden features of adaptation in evolutionary history, there's lots of traits that are simply not adaptive. And one of the most effective critics of this whole view, Stephen Jay Gould, introduced a term about 30 years ago that summarized this view. Gould was a paleontologist at Harvard, and his thinking will come up a whole lot in next week's lectures. He teamed up with another guy, a molecular geneticist at Harvard named Richard Lewontin, and critiquing a lot of this, they came up with a term saying an awful lot of traits have evolved that exist not because they're adaptive, but because they're spandrels. So at that point, everybody in the field had to go and run to their dictionary because Gould and Lewontin were all being snotty and coming up with fancy, highfalutin culture terms, because it turns out spandrel is a term from architecture. So immediately they already won points off of everyone else for being all cultured and stuff. But a spandrel is you got some medieval building. Remarkably enough, that's a medieval building. And it's a nice medieval building. And what you get is you have these arches that there winds up being a space between arches. There's a space between arches just because you can't build arches next to each other without there being like the space here. This is solid brick or Lego blocks or whatever. So you have these arches and there's a space in between and you got the space. It's not there for any good reason. It's there because you can't put two arches next to each other without coming up with this triangular space. And the architectural term for those triangles are spandrels. And spandrels in architecture is something that forms between two arches. And as Gould and Lewontin use the term, a spandrel is some trait that occurs simply as the unavoidable outcome of some other traits that are being selected for. You can't put two arches together without getting some sort of triangular shape in between. And throughout history of architects making spandrels, these would be artistically built up and all of that, and there would be all sorts of decorative stuff on that. So if you are an adaptationist folly person, everything has an adaptive purpose, you look at that and, oh, there's got to be some reason why spandrels have evolved and have these elaborate secondary sexual features on spandrels of little gossamer things flying around and stuff, and there's got to be an oh, I know, because people in church will look up at it a whole lot and their necks will get sore, and when their necks stop being sore, they will decide this is the glory of God and they will increase their belief for some. That's why architects put in spandrels. No, you can't build buildings like this without spandrels happening, and just because they were there, why not? Let's decorate them. That's the evolution of spandrels. And in Gould and Lewontin's critique, an awful lot of stuff where the sociobiology types would sit there and say, we're going to have a just so story contest now and use our principles and figure out some way in which this is actually adaptive. He's saying this actually wasn't selected for because it's adaptive. It was an inadvertent byproduct of something else. So here's an example that would be given. Humans have chins. Humans have chins. Apparently all humans do have chins hidden away someplace or other. Humans have chins, and weirdly, we're like the only primate that does. You look at like other apes and stuff, and they have this sort of weak chins that suggest sort of immoral characters and criminality and things like that. We're the only species that has chins. And apparently, there's been some like nutty adaptationist school somewhere back when trying to make sense of why it is that humans evolved chins, why it is that human faces come to a point there, and what's the adaptive advantage, and you can like stab rivals, or you can get crumbs from out of the corner on the floor and stuff with your chin, until somebody figured out that there's no way that you can have a primate face that has a muzzle foreshortened and a jaw at this angle. You do this and you do this, and you're going to get this little spandrel thing sticking out there, and oh, there hasn't been selection for a chin because you have selection for a hominid face with a shortened muzzle. This thing pops out there, and suddenly all those doctoral theses about the reproductive advantages of chins go down the tubes there. It was merely a spandrel. And Gould and Lewontin's argument is that there's lots and lots of spandrels, that this emphasis, everything is adaptive, everything is the outcome of competition that's produced that. An awful lot of stuff is excess baggage that merely gets carried along, because the evolutionary process isn't all that efficient. You're starting with a mollusk if you want to make yourself a squid. Giving rise to another concept, another term in the field, André Lewaf, who was a French, it wasn't Jacques. There were these two French molecular biology guys, and they both got Nobel Prizes, and one of them said this, and I don't remember which, but I'm going to say it was Lewaf who did, because I like saying Lewaf. André Lewaf once said that evolution is not an inventor, evolution is a tinkerer. It works with pre-existing structures, and the notion that you're going to see this everywhere, you're gluing stuff together, you're duct taping this part of the pelvis to this part of the circulatory system, whatever, and you're going to have spandrels. An awful lot of what those folks go through in these whole song and dances of adaptation are merely spandrels. Evolution is not about optimizing every single trait. What comes out of that is this notion that an awful lot of this world of competition really isn't about competition. You stick with these models, the models we've been covering for the last 2 days, and every bit of advantage is going to increase the number of copies in your genes. What's the only possible outcome? You need to compete, you need to outcompete. It was a critique of this whole view as being one driven very strongly by competition, because all the traits there are driven by having to out-reproduce the next individual. Another realm in which this has been critiqued has been by, interestingly, evolutionary biologists from the Soviet Union. And where what was emphasized there in Western sort of emphasis in evolutionary biology, most of evolutionary selection was about competing with other individuals. Sexual competition, competing for running away from the predator faster than the guy next to you, competing for food, all of that. If you're sitting there in the Soviet Union, you're thinking about evolutionary biology, what you're mostly thinking about is evolving the means of surviving climate demands, surviving extremes of climate, what would be categorized as abiotic rather than biotic, abiotic demands in the environment. And when you look at species where most of their survival is about surviving ecological extremes, environmental extremes, you don't see a whole lot of competition. That's a world with far less. And throughout the years, the Soviet evolutionary biologists tended to emphasize abiotic selection, which had far less of an emphasis on competition. The final piece, though, the gradualism. The gradualism is very intrinsic to this, because if everything is adaptive, every little bit of advantage is worth fighting for, and competition and killing each other's infants and killing each other's sperms and oviducts and all of that, and every little bit is going to make for a little bit of an advantage, and you will have this gradualist change. And the huge challenge that came to that was from Gould and another scientist in the 80s, a notion that maybe that's not what evolution looks like. Maybe evolution is not occurring in these gradualist incremental steps. Maybe what occurs instead is long periods of nothing happening, and then a rapid evolutionary change. Rapid evolutionary change, something that they called punctuated equilibrium. And the whole next lecture is going to be about this, critical implications. The genetics of how stuff evolves turns out to fit far better the molecular genetics with these models of punctuated rapid change and stasis, equilibrium, punctuated change, rather than these gradualist models. And if that's what it looks like in these steps, most of the time, you're not punching it out for every 1/2 of 1% reproductive advantage, and it's all competition. Most of the time, you're not punching it at all, and there's periods of severe selection, and then long periods in which nothing happens. A step function like that, the role of competition is reduced tremendously in thinking about evolution. So we're going to spend a ton of time looking at the punctuated equilibrium model to give away the punchline that I know you are all on the edge of your seats for. I kind of think it's right in a lot of the circumstances of evolution, because it fits much better with the molecular biology. Finally, In addition to these critiques of show me the genes, everything doesn't have to be adaptive because there are spandrels, and besides, this is really wimpy science where science counts as making up the best story, and there's no reason to think that it's gradual. There's mechanisms for this as well. In addition to that, there's always been a final realm of criticism of this style of thinking, which has been a political critique. And in lots of realms of science, you can't really imagine a political critique as to whether cilia on amoeba flip this way or that way, or a critique as to whether like plants should have 3 clover things or 4 clover things, that there's not a political factor in that. There's a huge amount of socio-political implications that run through this entire field, because it addresses issues of like how natural, how evolutionarily ancient, how evolutionarily honed, how adaptive are things like whether a species, a society has male domination or not, has strict hierarchies or not, has certain degrees of aggression, has certain patterns of sexual coercion, and the whole world arguing that these things have a naturalistic grounding and adaptation versus a whole world that says that's gibberish, most of the time nothing's happening, and when it does, a lot of it is spandrels. Those have very different political implications. And from day one, the critics of this entire world have made a point of something that initially seems kind of silly, but may not be silly, which is the entire founding generation of these sociobiologists were white Southern males. Make of that what you will. E.O. Wilson from Alabama, Robert Trivers, Irvin DeVore, a whole population of these folks, the first generation of these people were all white males from the American South. Whereas Gould and Lewontin, all of these guys, were Northeastern Marxists. Okay, so we got kind of a major contrast here, and the critique of the sociobiological view is, isn't it interesting that their notion of how evolution works just happens to emphasize the naturalness of a system that rewards them for the inequalities that we have? Isn't it interesting that their models predict the naturalness of a world in which they're the ones who benefit most from the notion that this is natural? And from day one, that has been a strong critique, and that's not like critiquing cilia amoeba theories for political implications. That is a very real one, because the notions that this is pertinent to making sense of, is rape a human psychopathology, or is rape a competitive strategy? Is the fact that children are more likely to be killed by stepfathers than by biological fathers, does that represent something about the typical socioeconomic pressures in families with step-parents versus the natural biology of gene competition through competitive infanticide? This has a lot of implications. And from day one, as this field emerged in the late 70s, there was a huge political agenda that was assumed to be there, sufficiently so at one lecture, famous conference in 1977, where E.O. Wilson was presenting there, a bunch of people rushed the stage, knocked him off the stage, and dumped water on him, and chanted whatever it is in German, saying, "We will have a society of law and order." This is pretty agitated circumstance for thinking about science. This was a group in Boston called Science for the People, which is a Marxist group at the time there, and saying all this sociobiological stuff is doing is justifying a world in which it is male-dominated, stratified, and where aggression and competition pays off. And this has been highly controversial from the beginning. The one counter to it, and I think a lot of that is valid, and I think a lot of it instead defaults into models where this is not making a whole lot of sense. The one thing to be pointed out is when we see what punctuated equilibrium is about, it's exactly the sort of world of evolution that produces the sort of world that a Gould or Lewontin would want it to be. One which minimizes competition in favor of cooperation, and a whole pattern of stasis and rapid dialectical change. It's exactly the world that two Marxist geneticists would say evolution should be about. So I think as a first pass here, seeing this stuff is dripping in socio-political implications. So starting Monday, we will look at this punctuated equilibrium, taking apart this piece of the story, and basically for the rest of the course, examining.
For more, please visit us at stanford.edu. Stanford University.
So we pick up from whatever the last day was, and we do our first disciplinary leap as promised at the very beginning of the course. The entire function of the first half of the course is to leap from one bucket to another, such that just as soon as you feel like you are getting mildly comfortable in one, we will spend time trashing it from the standpoint of a different discipline. This is our first one of these. And the whole point of this jump is to show on a certain level exactly where one discipline's notion of an explanation ends, another one begins. And what this one will be about is how evolution works down on a molecular level. Where did we leave off after finishing our overview of the evolution of behavior? We finished with a bunch of criticisms at the end, criticizing sort of what some of the basic tenets were of that view. First off, that notion of heritability. Heritability, assuming that all sorts of behaviors have a genetic component, have a genetic basis, have a genetic cause, and we will see those worlds of differences in those words there. And what we saw was that that could be the case, that could not be the case. That's going to be what we focus on a great deal today. Another one of the basic tenets, this notion of adaptation. Everything you see is wonderfully adaptive. The outcome is exactly the process that evolution brings about to optimize results. The counter view being the world of spandrels. A lot of the time, stuff gets carried along as baggage. Things evolving aren't necessarily things that have been sculpted into being as adaptive as possible. Another critique, and one that's also central today, is that whole emphasis on the gradualism, on small incremental changes. And what I alluded to at the end the other day is going to see a viewpoint where something very, very different is proposed to be going on. One that will make sense only eventually when we see some pretty unexpected things about genetics and the molecular biology. Okay, so starting off, what we also finished with was a notion that there are political agendas that run through every aspect of this subject, and if that very first lecture back when talking about how some of these viewpoints influence who gets a lobotomy, who winds up exterminated, who winds up being viewed as educatable or uneducatable, etc. Political views run throughout all of this. You will see very strongly in this realm. Okay, so that viewpoint of the sociobiologist, evolutionary psychologist the other day, one of the tenets going after the notion of heritability. The notion that these behaviors are heritable, are genetically influenced, blah, blah. How would folks in that business do it? What they would do is exactly what we were seeing, which is say, okay, here's some behavioral structure. You know how evolution works and evolution of behavior, and individual selection, and kin selection, reciprocal altruism, and evolution of, and all the above. And then they say, well, with that framework, that explains the behavior pretty well. Go show me something better. Show me a better theoretical structure that's even more explanatory, more predictive, and until you come up with something better, I'm going to assume that this one is right, and this one comes with a large assumption of heritability of genetics. All built around inferentially. These are highly structured models built around how genes work. They explain what we see with these behaviors, and until you can give me a model that does even a better job, I'm sticking with this, and this counts as my evidence for a genetic component to this set of behaviors. What we'll see is that's exactly where the molecular biologists decide that they finally have gotten free of people doing, I don't know, poetry for all the science, and my God, that counts as science, coming up with a bunch of these rules there and saying until you come up with fancier rules or a more pleasing just so story, I win. That counts as science. Complete contempt for this approach. This is where a more molecular stance about it all takes off. And what we'll see that not only will have lots to say about adaptiveness, it will also have lots to say about gradualism. Okay, so starting with it, in order to make sense of this, for a molecular biologist, what is evolution about? When you see traits that have evolved, what's it about? By necessity, well, one is immediately talking about our genes. Genes, in this case, not as some constructs that one wants to maximize copies of and ones that your cousins have only whatever percentage of in common with you, but genes instead molecules, genes as information, genes as strings of DNA. And I'll assume by now there's like a basic level of shared knowledge about this after hearing that a lot of folks came to the catch-up session last week, which I think is a good idea. But what is all built about, all of this notion of genes eventually way out the other end having something to do with behavior is the intermediary of proteins. Proteins are important. Proteins are important, not just to have in your diet and make you run fast and strong and all. Proteins are in lots of ways the structurally most important things you've got making up cells. Proteins have endless roles. Proteins hold the shapes of cells together. Proteins form messengers, hormones, neurotransmitters, all of this. Proteins are the enzymes that do all sorts of the most important stuff. Proteins, et cetera, et cetera, et cetera. Proteins are the workhorses of what has cells doing what they're supposed to do. So the question, of course, becomes what codes for proteins? And this is where genes come in. Our basic issue here is this flow of information. Genes specify proteins. Proteins are made up of constituent building blocks, amino acids. There's approximately 20 different ones that are commonly occurring, and each one has to be coded for with a different DNA sequence. A different DNA sequence of 3 letters, 3 nucleotides. And I hope I'm not hitting a level here where people for whom this is new, this is going way too fast, and everybody else, this is way too boring, but hang on for a while in that case. Okay. So DNA codes for amino acids. A long string of DNA coding for a sequence of them will code for a sequence of amino acids which get plugged together, and you then have a protein. You got one intervening step, which for our purposes is like not really interesting and we could ignore for the most part, which is there's an intermediary step. Genes at the level of DNA, sequences of DNA, first specify an intermediate form called RNA, and it's from that that you get the readout forming the proteins. Now, everything about the function of this is built out of the following sequence. If you know the sequence of DNA, you will know the sequence of RNA. You will know the amino acid sequence. You will know the behaviors. And what we'll see is that's exactly where the molecular biologists decide that they finally have gotten free of people doing, I don't know, poetry for all the science in it. My God, that counts as science, coming up with a bunch of these rules there and say, until you come up with fancier rules or a more pleasing just so story, I win. That counts as science. Complete contempt for this approach. This is where a more molecular stance about it all takes off. And what we'll see that not only will have lots to say about adaptiveness, it will also have lots to say about gradualism. Okay, so starting with it, in order to make sense of this, for a molecular biologist, what is evolution about? When you see traits that have evolved, what's it about? By necessity, well, one is immediately talking about our genes. Genes, in this case, not as some constructs that one wants to maximize copies of and ones that your cousins have only whatever percentage of in common with you, but genes instead molecules, genes as information, genes as strings of DNA. And I'll assume by now there's like a basic level of shared knowledge about this after hearing that a lot of folks came to the catch-up session last week, which I think is a good idea. But what is all built about, all of this notion of genes eventually way out the other end having something to do with behavior is the intermediary of proteins. Proteins are important. Proteins are important, not just to have in your diet and make you run fast and strong and all. Proteins are, in lots of ways, the structurally most important things you've got making up cells. Proteins have endless roles. Proteins hold the shapes of cells together. Proteins form messengers, hormones, neurotransmitters, all of this to come. Proteins are the enzymes that do all sorts of the most important stuff. Proteins, et cetera, et cetera, et cetera. Proteins are the workhorses of what have cells doing what they're supposed to do. So the question, of course, becomes what codes for proteins? And this is where genes come in. Our basic issue here is this flow of information. Genes specify proteins. Proteins are made up of constituent building blocks, amino acids. There's approximately 20 different ones that are commonly occurring, and each one has to be coded for with a different DNA sequence. A different DNA sequence of 3 letters, 3 nucleotides. And I hope I'm not hitting a level here where people for whom this is new, this is going way too fast, and everybody else, this is way too boring, but hang on for a while in that case. Okay, so DNA codes for amino acids. A long string of DNA coding for a sequence of them will code for a sequence of amino acids which get plugged together, and you then have a protein. You got one intervening step, which for our purposes is like not really interesting and we could ignore for the most part, which is there's an intermediary step. Genes at the level of DNA, sequences of DNA, first specify an intermediate form called RNA, and it's from that you get the readout forming the proteins. Now, everything about the function of this is built out of the following sequence. If you know the sequence of DNA, you will know the sequence of RNA. You will know the amino acid sequence. You will know the protein that thus is made. You will know the shape of the protein, and you will then know the function of the protein. And that is this critical link between what DNA, genes, evolution, blah, blah, are about, and the actual things that pop out in the other end and do something. For the very critical reason that everything about protein function is built around shape. Here is a cliche that is required by law to be said at this point, which is all sorts of effector proteins fit into other molecules, other effector proteins like a, okay, everybody say it out loud. Everybody know this cliche? It fits in like a, yes, okay, cliched education at its best, like a lock and key. For those of you who are not tortured with this one from early on, it's the whole notion that a shape of a protein imparts information insofar as it interacts with something else of a shape which modifies it, which complements it, whatever. The whole world that we will have of various hormones and neurotransmitters will consist of hormones and neurotransmitters going into receptors where there's a critical relationship in the shape between the messenger and the receptor, and all of this is driven by proteins, protein shape, etc. fit into other molecules, other effector proteins like a, okay, everybody say it out loud. Everybody know this cliche? It fits in like a, yes, okay, cliched education at its best, like a lock and key. For those of you who are not tortured with this one from early on, it's the whole notion that a shape of a protein imparts information insofar as it interacts with something else of a shape which modifies it, which complements it, whatever. The whole world that we will have of various hormones and neurotransmitters will consist of hormones and neurotransmitters going into receptors where there is a critical relationship in the shape between the messenger and the receptor, and all of this is driven by proteins, protein shape, etc. All of this is driven thus by DNA sequences. Shape is everything with this. What you get from that is, of course, the question of where do you get different shapes from? And those 20 amino acids, for our purposes, for the purposes especially people without a strong chemistry background, all that's pertinent here is the 20 different amino acids have different degrees of being attracted to or repelled by water, which most proteins are spending their life swimming around in water. They are pulled towards water, they avoid it, they are hydrophilic, hydrophobic. All that means is different amino acids gets pulled into different positions by their relationship with water, and thus a string of amino acids, the shape it winds up getting with is determined by that amino acid sequence. And there's a whole world of really interesting, horrible neurological diseases called prion diseases, which show that everything I just said is wrong, but for our purposes, everything I just said is right. So this is where you get this critical relationship. Know the DNA sequence, know the coding, and out the other end, you will get protein shape because of this business of different amino acids having different relationships with water, and thus strings of them coming up with different three-dimensional structures, and out of that will coming function out of the famed lock and key nature between shapes of functions and shapes of other things. Okay, just once again for people fairly new at this, no diagram there. One of the most interesting, or the blank slate, one of the most interesting things that proteins do is when they are enzymes. Enzymes, everybody knows that enzymes are important because they put them in your laundry detergent and advertise about how cool it is that you have enzymes in your detergent. But what enzymes do is they catalyze reactions. They cause reactions to occur, which left on their own would be very, very rare events. What enzymes do is accelerate vastly, a gazillion times over, the speed with which these happen. Catalyzed reactions, what do I mean by that? For our purposes, they can take two things that aren't connected and stick them together, or they can take one thing and break it apart. For our purposes, that's what enzymes do. Virtually every enzyme out there is a protein. So proteins fit into other things and send out messages. Proteins are enzymes, pull things together, pull them apart. Proteins are structural. Again, a whole lot of the superstructure of your cells are held together by proteins. This is the realm of what they do. Notice here, suddenly there's a change of shape in a protein if it's an enzyme, because what's it doing? In some way, it's got to be pulling apart something or putting something together. The shape in some cases also can change as a function of what this protein is doing with its job. We will see a classic version of that are channels. Channels in which chemicals can flow in or out of cells, ions for chemistry types, channels that will open up under some circumstances, close under others. So protein structure not only gives shape and function, but it gives the circumstances where the shape might change in a functionally relevant way. Okay, so out of all of this comes the central dogma of life, and this was proposed by Francis Crick of Watson and Crick fame. And Francis Crick was the one who formalized saying the central dogma of how life and information flows is DNA to RNA to protein. And that became defining. An entire generation of babies were told that at birth. This is the flow of information. It has been violated in all sorts of interesting ways, which will dominate a lot of what comes. But this was the central dogma. One way in which it's violated, and again, this notion is not DNA to RNA, but the notion of whatever your DNA sequence is, whatever the gene is, whatever structure of a protein it codes for, the flow of information is going to be from DNA, RNA to protein, DNA as running everything. Note the importance of that in the very statement of this as the central dogma, the canonical flow of information in life. It all starts with DNA. DNA is the one sitting here deciding when information is going to flow from DNA to RNA to DNA as knowing what's happening. And a lot of what we will see shortly is DNA knows squat. DNA is not making a whole lot of decisions there. But one simplistic way in which central dogma went down the tubes in the 1970s or so, and one that's tangential here, but just as a first blow against central dogma, one of the things that's interesting is there are things called viruses, and what viruses do for a living is get into organisms. Viruses, in the classical form, being little smidgens of foreign DNA, which are able to get into the DNA of your own, hijack the processes there, and make the cell function for its own parasitic, vicious needs. And so you're changing the starting step of how the central dogma of life, and thus you're going to change RNA and protein, all of that. In the 70s, it became apparent there was one weirdo viral world of viruses made out of RNA. This intermediate form. All sorts of people were extremely upset about this and tried to ostracize the scientists who came up with this and told them there's no way. But eventually what was shown in Nobel Prize winning glory was there's a class of enzymes. Enzymes, they pop up, a class of enzymes that could take the RNA information and turn it back into DNA viral information, and then it does its thing. Huge blow to the central, that here's information running from an RNA virus somehow being reversed back to a DNA form, and thus these are called retroviruses. Insert in the DNA, and off they go from there. So this was a major blow. Everybody eventually came to terms with this, but it still is a minor footnote in this Crickean world of everything flows from DNA. It is the Bible, it is the lawgiver, it is the Holy Grail, it is where it all starts. So stay tuned. Okay, so given that framework, one should immediately get mighty impressed with what if something changes in the DNA? What if one of the bits of coding is coded incorrectly? What if we now have on our hands a mutation? And what we will focus on here is classical realms of mutation in genetics and how that plays out in classical gradualist models of evolutionary change, and then see how all of that falls apart when you look at what's really going on. So starting off, what you can have, and again, this is going to be a review for people with a background in this, newcomers to this, hopefully this won't be going too fast, but broadly, when you were talking about this world of micromutations, for our purposes, what we will mean by a micromutation is when one letter in your DNA sequence of information gets accidentally miscopied, or gets changed by radiation, or gets changed by some chemical compound in the environment, some such thing, where there's a mistake and one letter in the DNA sequence comes up wrong. As I noted before, pairs of triplets. There we've got 3 pairs of triplets. And what we've got here is at the DNA level, an amino acid is coded for by 3 base pairs. A triplet, 3 of these, next amino acid coded for, next one coded for. So what we've just raised is the possibility of a mutation occurring. Something changing in one single one, and now asking what are the consequences going to be in classical realms of genetics and evolutionary change. Okay, broadly you can get 3 different versions of stuff going wrong. One is where one of these letters, nucleotides, translating, not essential to have that down, where one of these letters is accidentally changed into another letter, a point mutation. In some cases, point mutations are of no consequence at all. How can that be? For this very simple reason, going through some math here, there are 4 different potential letters. At each one of these sites, DNA comes in 4 different letter types, letter flavors, 4 different types of bases, and thus you can have 4 different ones in the first position, times 4, a total of 64 possible 3-step combinations of DNA letters. A possibility of 64 of them, and you're only coding for 20 amino acids. So you use up some of the 64 for signals saying stop or start or thank God it's Friday or who knows what. But what you have, though, is a large redundancy. You have a number of different triplet sequences coding for the same amino acid. There's redundancy in the genetic code. And in general, where you get that redundancy, where you get the differences in 3 triplets, a couple of different triplets that code for the same amino acid, they'll tend to differ in the middle one of the 3 letters. That's the easiest one to have a change, where it doesn't change the overall consequence of it. So you will tend to have redundancy. Each amino acid is coded for by a number of closely related triplets. So, first possible type of mutation, this point mutation, where one letter is flipped to another. Potentially, this could be of no consequence whatsoever. If you have flipped to a letter out of the middle one, which happens to leave you with a triplet that codes for the same amino acid. In that case, you have a neutral mutation of 0 consequence whatsoever.
Now we discuss DNA replication, and I think it's important to make the distinction that we're talking about replicating DNA. So we're using our DNA template to make more DNA, a duplicate copy of the DNA. And we're not talking about transcription and translation, which involves the movement from DNA to RNA to proteins. And so make that distinction in your mind, that replication involves making copies of the DNA, and this happens during the S phase of mitosis, for example. Whereas the central dogma of DNA to RNA to protein is what's involved with transcription and translation. So as far as DNA replication goes, there is a directionality to it that I think I'd like to discuss before we get to all of the individual enzymes involved. There are polymerases, there are ligases, primases, and so on. But before we get to that, it's important to discuss the directionality that this goes. And I think the best way you can remember it is by thinking of a studying strategy. When you study, you read up on something and you write it down. And what I mean by this is that the DNA polymerase will read up, so it reads in the upstream direction from 3 to 5. But when it writes a new strand, when it writes a duplicate of this template strand, it writes in the downward direction, so it builds it from 5 to 3. And what this means is that here's our polymerase enzyme, and as it moves along, it will be producing a new strand of DNA. And so it's going to read it in this direction, moving from 3 to 5. So you read up, you read from 3 to 5. But when it writes it, it will be writing and producing new segments that are complementary to our template strand. And these will be in the 5' to 3' direction. And so What that essentially means is that it's going to work with a template strand, and it's going to start from the 3 end and move toward the 5 end. But as it writes and produces this new strand, remember it's anti-parallel, and so it will start with the 5 strand and move toward the 3' direction. So read up and write down is a good way of understanding the directions through which DNA replication occurs. You read starting at the 3 and you move toward the 5, but as you write it, the new strand is being produced with from the 5' end over to the 3' end. And now we'll get into the specifics of the enzymes involved in the replication process.
Last week, scientists announced that they've created a weird synthetic DNA in the lab. They added 4 new letters to the usual 4, creating a molecule with an 8-letter code, which is pretty freaking mind-blowing. Having an 8-letter code really expands DNA's ability to store information, so this synthetic DNA could change the way we keep data long-term. And it makes us realize that our DNA isn't really special. For billions of years, all life on this planet has stored its genetic information using molecules of DNA. Strands of DNA pair thanks to hydrogen bonds to form a long spirally structure called a double helix. Each of those strands is made up of sequences of 4 different nitrogenous bases: adenine, guanine, cytosine, and thymine, or A, G, C, and T. And it's the sequences in which these bases appear that forms the genetic code underlying life as we know it. And yeah, I know, you learned about all this stuff in biology class, but the researchers publishing last week in the journal Science kind of literally rewrote the genetic code. They made DNA with A, G, C, and T, but also with 4 new bases, which they called P, B, Z, and S. The researchers called the new DNA Hachimoji DNA, from the Japanese words for 8 and letter, in order to show that Hachimoji DNA was legit and could really be used as a genetic code. The researchers had to be sure it could do 3 things: reliably form matching base pairs, keep its shape when mutations occur, and be read and translated into RNA. So they did lots of tests that showed that, similar to how A pairs with T and C pairs with G, P likes to bond with Z, and B wants to pair with S. So you end up with 2 complementary strands of DNA. And these bonds were reliable and stable no matter which other bases they were next to in the sequence. Then they looked at the effect of the synthetic bases on the overall structure of DNA. One important criteria of DNA is that the nucleotides need to all be the same size, so that they don't disrupt the double helix. If swapping bases into different parts of the code caused the whole thing to collapse, it would be impossible to have mutations. And since mutations are the foundation of Darwinian evolution, life can't really exist without them. So the researchers created 3 crystal structures of Hachimoji DNA with different sequences, and found that the synthetic nucleotides didn't warp DNA's famous double helix too much. And the last thing they wanted to make sure of was that the enzymes responsible for translating DNA into RNA could actually read the darn thing. After all, there's no point in storing information if no one knows how to read it, and DNA isn't useful unless it can be put into action. But enzymes the researchers tried kept having trouble with the S base, because it has a slightly weird chemical structure that messes with the strength of one of the hydrogen bonds it forms with B. So the researchers looked through variants of the enzyme and finally found one that could read all 4 of the synthetic bases and make an RNA strand when given the base's synthetic RNA counterparts. Which means that Hachimoji DNA works. And beyond the gee whiz of it all, there's good reason to get worked up about this new weird DNA. For one, it's capable of storing so much more information. If you only have 4 letters to record stuff with, you need longer sequences of those letters to make enough different combinations to represent different information. But if you have 8 letters, all those snazzy combinations mean you can store more info in less space. And we're not just talking about genetic data. Scientists have been experimenting for decades with the idea of using DNA as a kind of hard drive. It's already a pretty efficient data storage system, since it's really tiny yet stable enough to have a shelf life of thousands of years, maybe up to 1 million under the right conditions. Already, researchers estimate that 1 gram of single-stranded DNA could hold a whopping 455 million terabytes of data. Hachimoji DNA DNA could hold a lot more, depending on how the data is coded. The newfangled DNA could also have medical and biotech applications. For example, small strings of DNA can be used to create a kind of molecular probe called an aptamer. Based on the sequence, these aptamers can locate specific molecules, like the compounds that are only found in or on cancer cells. And that means they could help target medications or light up tumors so doctors know what to remove. Previous work from this research group has shown that DNA aptamers with Z and P bases are even better probes because more base options allows for more specificity. So it's likely Hachimoji DNA will be even better. And then of course, there's what this research says about life itself. I mean, it's not that we've created alien life forms or anything like that. Hachimoji DNA isn't self-sustaining and it can't exist outside the lab, so it's not really life. The research was funded by NASA though, in the hopes of better understanding what other life might look like if we were to find it somewhere out there in the universe. Because what Hachimoji DNA tells us is that there isn't something perfect and singular about the 4 bases we.
So that's not a very exciting mutation. In some cases, you can have a single point being changed where you wind up with a different amino acid coded for. And that's most typically if it's a mutation in here or here, rather than the boring middle one, you get a different amino acid. Is that the end of the world? Often not at all, or only minimally, because a lot of the amino acids have similar feelings and ambivalencies about water, similar responses and similar shapes. It won't be exactly the same shape, but the protein will function somewhat the same. So you could have, in this case, a point mutation, and most people would not find the second line to be impossible to make sense of in the context of the first one. Or you can have a point mutation, which by changing one letter produces a different amino acid, and this one is majorly different. This one has a completely different set of attractions or being withheld by what produces a protein of a very different shape and potentially a very different message. And shown here, just changing one letter in this case, one single point mutation, and you dramatically change the meaning of that. And I actually did that once in a grant proposal, sort of the final paragraph. We have now accomplished X, Y, and Z with your money over the last 5 years, and no wonder it didn't get renewed. So in that case, you see a single point mutation of great consequence. So you can have one of these letters, one of these nucleotides, mistaken for another, entirely neutral, moderate consequence, or major disastrous consequence. Second way of classical mutations, the second version that it could come with, is there's a deletion. One of the letters gets lost in the process. And what you then get is a frame shift over to the missing spot there, and the D from here now finishes up this. And what you can see is it suddenly becomes dramatic gibberish, and it would continue that way. A deletion mutation in classical genetics is major league. It totally changes the coding downstream from that. Third classical type, an insertion mutation, one where you now accidentally double the letter and you frame shift it in the opposite direction. Just as screwing up of meaning, major consequences there. So you have point mutations, which can have major consequences or can have none whatsoever and anything in between. You have point mutations, and then you have insertion and deletion. The last two tend to have big consequences. So all of this is built around one single base pair changing. And all of this is built around, thus, one single protein changing its shape. And thus, this world of micro mutations, single spots that are mutating, what this tends to do, and this is an important concept for what's to come, what it does is it changes how well the protein does its job. It changes the efficacy of that protein by changing the shape a little bit, by changing it dramatically, all of that. And we can see, back to our lock and key, where if thanks to a mutation, this has a slightly different shape, it will fit into the lock. slightly less effectively may stay in there for a shorter time before floating off and thus send less of a message. On the other hand, if you've got a deletion insertion that dramatically changes the shape of this, you will change how well this protein does its job. It won't do its job at all because it's going to wind up with a completely different shape and not fit in there whatsoever. What we have here is a world of mutations which are changing the function of one protein at a time. That is microevolutionary change. And that's what we're now going to see played out. When can this make a difference? This can make huge differences when it's in the realm where, thanks to a change in the shape, the protein is completely out of business. Two examples. One where you take out the function of a pre-existing gene. First one, there is this amino acid. For our purposes, that's going to derail us. For our purposes, there's a chemical that occurs in the body called phenylalanine, which has its uses, but you don't want the levels of it to build up too high because it can become toxic, damaging to brain cells, to neurons. And fortunately, there is an enzyme made of protein which turns phenylalanine into something safer. That's all we need to know about it. So you have a mutation in the gene coding for that enzyme. Not a fancy mutation, just something in one of these categories, a classical single spot point mutation, and what you got there is an enzyme that no longer does its job. And as a result, this phenylalanine is not converted into the safer form, builds up and lays waste to one's nervous system. And thus you have a disease, PKU, phenylketonuria. Very, very common genetic disorder, and this is one where the outcome is a small mutation that has completely knocked this enzyme out of business, this enzyme which was able to turn phenylalanine into something safer. This is not a subtle outcome. Have untreated PKU, and your reproductive success by the rules of evolution is going to be real down near zero. This destroys the nervous system very rapidly after birth. So that's dramatic. Here's another dramatic one. And this one, anyone who took BioCore, I always bring this one in as a big hormone crowd pleaser. But here's an interesting thing that can go wrong with any child you might eventually have. Okay, you've got a daughter, and she's doing just fine, and she's growing up just fine, and things are terrific, and, you know, around age 10 or 11 or so, some of her classmates are beginning to reach puberty. You know, that's on the early side for the Western average, but it's not outrageously early, not a big deal. By about 12, you know, statistically about half of the girls in her class have reached puberty. 12 is about the Westernized average these days. She hasn't yet, not a big deal. A year later, she still hasn't. Well, this is nothing critical, but it's getting a little bit on the late side. A year after that, 2 years after that, she has still not reached puberty. So at that point, you take her to a physician who examines her closely and figures out what's up. And at some point, the physician is probably going to have pupils dilate or some sort of weird autonomic response when they figure out what's up, and then very calmly, in a premeditated way, sit you down for a little talk afterward and inform you that your daughter has not started menstruating, has not reached puberty yet. Your daughter has not started doing this because you don't have a daughter. You got a son. This kid here, for these last 14 years, has actually been male, not female. You have what is called, no, I'm not going to tell you what, okay, it's called, especially since it's in the handout already. Anyone want to guess what it's called? It's called a TFM, testicular feminization syndrome, and you wind up with a testicular feminized male. These are individuals who genetically are male at the level of chromosomes, at XX and XY business. These are individuals who have testes way up in their stomach or whatever, never descended down. These are people whose testes make testosterone. Testosterone out the wazoo, tons of testosterone, enough testosterone to put like antlers on your testes or something, that much testosterone, and nonetheless, you're getting a female phenotype. You are getting a female external genitalia. You are getting female everything. Yep, question? No, that's the fancy term for it. And you've just given away the punchline, you creep. Okay. So they figured out in this disease there's an insensitivity to something. What could that be? Okay, go and say it again. Yes, okay. What you've got here is one of those simple little classical mutations, and what it does is it changes the shape of the androgen receptor, the testosterone receptor, and at that point it doesn't matter how much testosterone is floating around, those target cells are not going to listen. This is consequential. This is a major consequence. This is one of changing gender phenotype, and there is a long and at times absolutely appalling history of what has been done with individuals who have testicular feminization syndrome, what counts as the medically appropriate intervention. There's some horrifying history there that could be straight out of the first lecture in terms of some notions of what counts as normal gender behavior. Another version of this one's a little bit more subtle because in this case it's not wiping out the function of an enzyme, it's just making it a little bit less effective at what it does. And this has to do with a disease that is found in two different populations, slight variants on it. One is up in the Dominican Republic in the mountains. The other is in the mountains of New Guinea. Interesting similarity there. In both cases, some fairly isolated inbred populations. So that sort of has genetics written all over it. But in these cases, there's a problem with enzymes that make testosterone. So this is a whole other world. Instead of the enzyme, instead of the proteins, and thus the genes somewhere back there, that code for the receptor for testosterone, the receptor that responds to this messenger. Here, instead, it's back way up there in the testes, a bunch of these enzymes which go through a bunch of steps and make testosterone. Biosynthetic enzymes there, proteins, genes, once again. So you've got a mutation in one of those critical biosynthetic enzymes, and it's not a huge one. The shape is a little bit off. Its efficacy, the effectiveness with which it makes testosterone is down a bit, and actually it's down a lot. So here's what you wind up getting. You get an individual who, like before puberty, has extremely low testosterone levels, even far lower than you would see in prepubescent males, in most cases someone who's genetically male, someone who never saw a whole lot of testosterone during fetal life, way below the threshold for the testosterone having any effects. So the person is born phenotypically female, female in appearance, female in external genitalia, and not internal, back to those testicular feminized males. What you find there's a vagina which goes nowhere because there's no ovaries waiting somewhere above that. There's the testes, but the external genitalia is just fine. What I think these days is the most common sort of medical ethics advice on what's done with that is the physician explains what it is, and says for all practical purposes, you have a daughter who is perfectly healthy, who is going to have a long, happy, healthy life, and simply cannot reproduce. That's the only consequence that's relevant here, and there's been a whole other world of reconstructive surgery and all sorts of very interesting, horrifying history. Okay, so back to this one. Because of the extremely low testosterone levels, because at a time of life when there's not a whole lot of testosterone around in a fetus, in a young male, and now it's below threshold for causing anything. Along comes puberty, and thanks to a whole bunch of changes in the brain, signals go out so that testosterone levels now come roaring up into the usual impossible levels in the males. And what happens here is the levels don't go up as much as they should because that enzyme is a little bit on the slow side, but they go up enough to pass threshold for beginning to have an effect. And somewhere, right around puberty, this individual changes sex. This individual transmits, jumps ship, and goes from female to male as a result of these androgenic effects suddenly coming in. It's not a complete switch there, but it's somewhat of a transition. This is bizarre. Again, this is not something subtle. This is something changing completely here, and again, one single mutation. And in this case, not even a mutation wiping out the function entirely of some protein, as in PKU or testicular feminization syndrome, but now you got a protein that's just working differently, slowly enough that you get this very different picture popping out the other end. What's most interesting is you have this intersection between molecular biology and the bioendo consequences of this mutation, all of that, and the cultural context of it. Apparently, in both of these populations, people's kind of adapted to it. It's not a big deal, you know. Puberty, sometimes you get acne, sometimes you get a penis, and like people just kind of deal with that. That's just part of the whole process. And cultural accommodation to interesting biology. One final example of where you can have a classical type of mutation and another version of the very mild difference. And in this case, it is not a very mild difference producing an overt disease. In this case, it's just producing something that probably differs in you from the person sitting next to you, which is you have a neurochemical system, a system of chemical messengers in your brain. If this is new stuff, hang on, it will all be explained in a couple of weeks. But this is a class of neurochemical signaling that has something to do with anxiety. A chemical messenger which decreases anxiety, and we will learn plenty about that down the line. And there's this regular class, they're known collectively as benzodiazepines. Do not panic. If you've not heard that word before, and certainly do not attempt to spell it right because it's not possible to. So benzodiazepines. There are synthetic versions of benzodiazepines which people might take when they're feeling anxious, like Valium, like Librium. Valium is a synthetic benzodiazepine. So benzodiazepines are protein, and they have a particular shape, and you guessed it, there are thus benzodiazepine receptors, which could do that whole lock and key deal going on there. And what you have are differences, small single point, in this case, not mutations, but simply different versions that one letter can come in the DNA sequence specifying the benzodiazepine receptor. These are not mutations, this is just normal variability in this one particular spot. Can specify 2 or 3 different amino acids that function roughly the same, so that you're changing somewhat the shape of the receptor and thus changing somewhat how long the benzodiazepine stays in there and how long the signal is sent, and it's all very subtle. And what does this begin to explain? Individual differences in levels of anxiety. It's only one of a gazillion ways of explaining it, but it's part of that picture there. Here we have individual differences. This explained a very interesting finding. For decades, people often try to breed rats, different rat lines for different behavioral traits, and it makes it very useful models. There are alcoholism-prone rats. There are rats that have been bred for being smart, for being not as smart at spatial maze stuff, etc. And for years, there have been a number of rat lines that have been bred that were either high anxiety or low anxiety rats. Very useful for understanding things like, what are they useful for? I don't know, they're just cool to have around. Very useful for understanding the effects of stress or things of that. So finally, modern era of molecular biology comes in, and those high and low anxiety strains differ in the shape of the benzodiazepine receptor in many of these cases. Again, tiny little differences. Okay, so what we've got here is classical genetics at the molecular level. These tiny little changes in one single base pair at a time. Point, deletion, insertion, a world in which it may or may not change the shape, where it could change it dramatically and produce some pretty exciting dramatic diseases where you wind up with a different gender than your chromosomes say, ones where you get more subtle differences, you change gender at your 13th birthday, or ones where we're just for the first time beginning to look at the individual differences that flow out from stuff like this, not them and their disease, but the individual variability. All of this is in the realm of single base pairs being changed. What all of this now allows us to do is translate this world of mutational changes into what does this look like evolutionarily? What does this look like in terms of explaining patterns of evolution? And you will know exactly where this goes right now. This helps explain the classical gradualist picture of little bits of change at a time. Little bits because you've got one protein which now works a little bit differently, and thus you get to ask a sociobiology question by having testosterone having a little bit more of an effect or a little bit less of an effect by way of molecular changes in this receptor. Is that going to increase the number of copies of genes that individual has? Is it going to decrease the number it has? How's it going to fit into all of last week's science? And you will know exactly how that drill will run. Oh, if thanks to one single base pair changing, you now have a receptor which functions in a slightly different way, which makes this individual 1.5% more fertile than everybody else that they're competing against. You just wait long enough and come back, and everybody in that population is going to have that different version of it. We can run the logic there. We have variability, Darwinian variability, thanks to a mutation. We have differential fitness. We've just defined an adaptive difference somehow or other. There's a smidgen of advantage thanks to the slightly different shape. And thus we have selection changing distribution over time. The smidgen more effective version will become more common in the population. And what's also intrinsic in that is that these are slow little steps of change. This is gradualism. So gradualism is absolutely commensurate with everything we got last week, built around adaptation, competition. Every little bit makes a difference, because a 1% difference in number of copies of your genes run at enough generations, and that's going to be a big difference, and all of it changing in a very gradualist, incremental way. This has popped up in a number of domains and could be really useful because it allows you, for one thing, to trace evolutionary history by looking at the changes of single base pairs, and that's allowed for looking at one really interesting gene, which we may talk about down the line, a gene called FOXP2. FOXP2 has something to do with language. FOXP2 was first identified in a family, all of whom had some sort of language communication problem. And people to this day argue whether it was mostly about coordinating the motoric aspects of speech, or was it something about grasping the symbolic message aspects of language, all of that. In any case, a mutation was found in this gene, and a whole cottage industry has emerged since then of studying FOXP2, because versions of FOXP2 occur all throughout the animal kingdom, in birds and rats and non-human primates and all sorts of things. FOXP2 popping up all over the place, and in all these different places it's got something to do with communication. It's got something to do with bird song. It's got something to do with rat ultrasonic vocalizations. Rats are constantly jabbering at each other in a range that we can't hear, and FOXP2 has something to do with in all of those, and different versions of FOXP2 in all these different species. And what becomes most pertinent is when you look at those differences, you will see tiny little differences in this tree across all these different species. One base pair difference between rats and mice, one base pair difference between hawks and elephants, that sort of thing. And then you look out at humans and a whole bunch of changes. A whole bunch of changes in a very short evolutionary period. What this suggests is, no wonder this is producing some very different stuff than these guys, a very different gene. And a very different gene people now have been able to back calculate due to a series of single base pair changes somewhere in the last 250,000 years or so. And that makes a difference. Totally cool, unsettling experiment that was done a few years ago, which was some people taking the human version of FOXP2 and knocking out a mouse line, knocking out their own FOXP2 and sticking in the human version and seeing what happens. And it was very interesting. They immediately sing the theme song from all the Mickey Mouse cartoons. Can they prove that? Okay, what you see is you got more complex types of ultrasonic vocalization in these mice. That's really interesting, and there's years of stuff to sort out what's going on with that. For our purposes, the main thing is here you have wound up with a very different world than chirping and buzzing and barking and all of that, and you wind up with a very different version of this gene where you can trace out how many amino acid changes it took, and every step of the way you were having some gradualist process. What also this allows you to do is look at a footprint, an echo of what the selection was like. Okay, important concept here. So back to that business, there's 64 different ways of coding for amino acids. A couple of them are just informational, stop messages and stuff. There's about 60 different ways of coding for 20 amino acids. So on the average, each amino acid could be coded for in 3 different ways. So suppose you throw in a random mutation, and of those, what you find is of the 60 possible mutations, 40 of them will not cause a change in an amino acid. Statistically, 2/3 of the time, there will not be a change. So in other words, if you scatter a whole bunch of mutations and you wind up seeing 2/3 are neutral in terms of their consequence, and 1/3 actually causes a change in an amino acid, that's telling you it's happening at the random expected rate of mutations popping up that are either consequential, changing an amino acid, or inconsequential, just coding for a different version of the same amino acid. Now suppose you find a gene that differs, and you look at the ways in which it differs, and 99% of the base pair differences over the course of this 5 million base pair long gene, 99% of the differences make for a different amino acid than beforehand. In other words, at a much higher than expected rate if this was a random process. What is this an echo of? Of very strong selection, of very strongly advantageous traits driven by these changes. Term used, this would be a mark of there having been positive selection for this trait. In other words, the changes that it has gone through over the gazillion years has not been just by random mutational rates of neutral, and remember every amino acid is coded for 3 ways. If 99% of them are consequential, there has been some major hard-ass selection going on there. Alternatively, if you find a long gene with a whole bunch of mutations and 99% of them are neutral, make no change at all, what does that tell you? This protein's function, you do not want to mess with it in the slightest, even a minor change in the function of it, and you don't pass on copies of your genes. This would be stabilizing selection. Negative selection, strong selection to make sure that this gene and its function downline as the protein does not change. When you look at the number of changes, the burst of mutations over the last 250,000 years or so that differentiated FOXP2 in humans from these other species, it's almost entirely evidences of positive selection. This did not just happen by chance. Every step of coming up with these new versions, these new amino acids in the sequence there, clearly were ones that were positively selected for. A lot of selection brought about this huge difference. Okay, so that begins to give a sense here of how these little changes can occur. At this point, we have to go through one of the all-time confusing things in genetics out there, which is something 2 factoids and 2 sound bites that seemingly totally contradict with each other. You share, on the average, 50% of your DNA with a full sibling. You share 98% of your DNA with chimpanzees. What's up with that? That seems a little bit unexpected. And those are 2 sound bites that everybody knows. Everybody knows it goes through like basic Mendel, 50%, 100% with an identical gene, identical twin, 50% with a full sibling. You know that song and dance by now. And meanwhile, one of the great sound bites of our evolutionary history and the mark of evolution and what in hell are you creationists thinking is the fact that, oh, humans share 98% of their DNA with chimps. This doesn't make any sense. 1 minute here to be spent on just clarifying that one that is not contradictory at all. Genes specify by way of proteins, and now this is taking a bunch of leaps down there. Traits, aspects, genes specified, to be totally simplifying here, genes specify for antlers, for dorsal fins, for petals and pistils and stamen, for kidneys, for just totally simplifying. So right off the bat, you will have some genetic similarities between two different species in that both of them will have genes that code for dorsal fins. You're thinking about two different species of whales. So they share genes coding for dorsal fins. Whales have dorsal fins. Neither we nor chimpanzees have genes for dorsal fins. Meanwhile, we have genes coding for a pelvis that is a certain shape, which either in chimps predisposes towards being able to walk bipedal for certain lengths of time. Us as well, we don't find these genes in apple trees. They don't have pelvises that are shaped like those, so they don't have, so when you look at the human and the chimp genome, 98% of the genes code for similar kind of things. We share with chimps an absence of genes for antlers and trunks and tusks and wings or who knows what. And we share all sorts of other genes in common having to do with our shared immune systems, et cetera, et cetera. So 98% of the genes code for similar types of things. So that's where we get the 98% similarity with chimps from. For each one of those genes, it could come in a couple of different flavors, a bunch of different flavors, and thus asking, not do both you and your full sibling have a gene for opposable thumbs, do you and your sibling have the same type of gene for opposable thumbs? So suddenly that's a different world of variance on each type of gene. So when talking about the amount of DNA shared in common, the genes shared in common with another species, what you're talking about are the types of genes coding for the types of traits. When we're talking about, from last week's notion, 50%, 25%, 12.5%, one brother or 8 cousins, what you're talking about are different versions of particular genes. Important clarification. Okay, so final thing here before the break, which is once again, notice with these models, these mutations where you can get slight differences in function, and thanks to a 1% difference in fitness and number of copies of genes, you get these gradualist changes. What's intrinsic in that, and where we get the political theme coming through, is thus there's competition everywhere. In terms of the evolution of behavior, the evolution of species, every little bit of genetic advantage will play out in some competitive way, in some little bit of an increase in reproductive success. Okay, whoa, there's a lot of apples there lit up. Well, that wasn't very subtle. Let's take a break. Whoever did that. Okay, 5-minute break. Are you slightly more fit by the rules of your population's natural selection, sexual selection, whatever, even if it gives you a tiny advantage thanks to this tiny change in this tiny gene, enough generations go by and that trait is going to become more prevalent. Final point there, intrinsic in this, is if every little bit of difference matters in terms of fitness and gene distribution, within the realm of behavior, every little bit of competition matters. A very, very intertwined sort of political, philosophical stance in gradualism as it applies to evolution. Okay, so that's been sitting there around forever. And in the 1980s, suddenly a very different model emerged. And this came from Stephen Jay Gould, who we heard about the other day. Stephen Jay Gould, and the person, the poor schnook who was always lost in this, another evolutionary biologist named Niles Eldredge, who somehow did not quite have the press that Stephen Jay Gould did, so he is lost to history, except for people who know what he's up to, and he's an amazing scientist. But Gould and Eldredge came up with a very different model. And I alluded to it the other day, and just drew it right there, and their notion was that gradualism is nonsense. There are not gradualistic incremental changes. Evolution is not being driven by small gradualist changes. Instead, what their model was is that there's long periods of nothing happening, of stasis, Long periods of nothing happening. If there's changes in DNA sequences thanks to mutations, they're not consequential, or if they're consequential enough to change the fitness of one organism 1%, that's not going to make a difference. Most of the time, no change is occurring, and when it does occur, it is incredibly fast, explosive periods of change followed by a new period of stasis. Evolutionary change comes in step functions rather than smooth gradualism. Long periods of stasis followed by dramatic jumps of evolutionary change in short periods of time. Thus, they call this the notion of punctuated equilibrium. Long periods of equilibrium and stasis punctuated by periods of very rapid change. And as I mentioned the other day, Gould was a Marxist and felt that Marxist sort of stance was running through all of the ways to think about genetics. And I don't know what's up with Miles Eldridge, but that was the case with Gould. And when you look at this model, this is like classically fitting with like stasis and revolutionary change and dialectical materialism and stuff like that. The last sentence, I have no idea what I just said. But apparently that's got something to do with that stuff. And once again, we see in a very different way a political theme running through a different worldview of what evolution is about, punctuated equilibrium. Okay, so where did the idea of punctuated equilibrium come to these guys? Mainly because Gould was not a biologist. He was certainly not an evolutionary biologist. What he was a paleontologist. He studied fossils. He studied the history, the evolutionary history of fossils, and apparently, like totally separate of his large theoretical models, he was like the world's expert on the evolution of some Caribbean snail shell over the last 10 billion years or something. He's one of those paleontologists who traces lineages of evolutionary change over the course of time with fossils, fossil records as the readout. So he's a paleontologist slash geologist in some ways. And when you do that, you notice something, which is you got gaps in the record. You've got your famed missing links. You have gaps in your evolutionary record there of what the fossils look like. And you're measuring some trait or other in these snails or trilobites or whatever you're looking at. And at this time period, the trait looks like this, and at this time period it looks like this. And this looks like a perfectly good gradualist model. And what Gould would notice is as the field got more and more information, more and more intervening steps on a lot of these fossil histories, they would start to look more like this. And every now and then, you would see something like this in between. And from that began to suggest to him this model of punctuated equilibrium. Most of the time, as assessed by the fossil record, nothing dramatic is happening, long periods of stasis. And what allowed this to occur is that for some fossils, you have incredibly detailed evolutionary history where you can begin to fill in lines, and they wind up looking punctuated in this way. So out of him comes this whole theory that it's all about punctuated equilibrium rather than gradualism. Right off the bat, what are the consequences of that? Little genetic changes don't matter. Competition driven by the notion of little changes mattering aren't actually occurring. A model saying most of the time, all of the notions of if you figure out the right kid to kidnap when the big guy is coming at you and you'll leave more copies and figuring out exactly who to be infanticidal to, all that, it's not going to make a difference in terms of gene distribution. Evolution is not being driven by that. And out of it came this very strong indictment of the sociobiological view of what you got there is a world where it's all about competition, where it's all about hierarchy, where it's all about domination, where it's all about that. Hey, isn't that interesting that that's exactly the sort of world that these folks live in who are benefiting from this, who started this theory? Very different notion here. Competition, selective advantages, all of that, most of the time, nothing's happening. So not surprisingly, all of the evolutionary types from last week did not like this one bit. And this was attacked left and right in some extremely valid ways. First form of attack is a very simple problem there, which is that you have two different disciplines happening here. You get a paleontologist and you get a evolutionary biologist, and they're functioning in completely different universes. Okay? Stomach problems, and they're functioning in completely different universes there. What counts as fast for a paleontologist? These are tens of millions of years going on. Whoa, incredibly fast evolutionary change going on there. That's like 100,000 years. That's like, are you kidding me? Said the biologists, the ones who study one generation at a time. That is asinine, that is ridiculous. This is them imposing models where this has absolutely nothing to do with how evolution actually works. These geologists got completely thrown off, and these paleontologists, led by Gould, simply orders of magnitude different scale of time. Yeah, maybe in some rough approximation of what they looked at, but they're not studying evolution because they're not biologists. Next critique. The next one made lots of sense also, which was you're not just an evolutionary biologist, but you're one who thinks about the evolution of the brain or the evolution of skin melanism, or the evolution of eye color, or the evolution of how many chambers in your heart you're going to have, or the evolution of any of these things that will leave no record whatsoever in the paleontological record, because all paleontology is about is shapes of stuff. Fossils. Fossils do not tell you what kind of brain was going on inside that fern. Fossils do not tell you anything about internal organs. Fossils do not tell you anything about behavior. So at this point, all of the evolutionary folks of the school from last week attack and say, yeah, what do they expect? They're studying the most boring possible things, the morphology of organisms. Ooh, just because the fact that humans over the last million years or so have not evolved sort of getting rid of the large trunk and roots that they have during springtime, and now they don't have them. Oh, that morphological change didn't occur, so obviously there's been stasis. Give me a break. What is interesting about evolution and evolutionary change, paleontologists can't pick up because all they can study are forms, morphology. So that was a big attack on these folks. So you've got the Gouldian folks, the punctuated equilibrium people, saying when you look at the fossil record, it's not gradualism. And we've got some really complete ones. And to this day, the majority of fossil pedigrees where there are very, very complete records show patterns of punctuated equilibrium. And back come the rejoinders. This is ridiculous, the time span they talk about.
I'm Rachel, hunter and slayer of fiends.
So that was a fiend.
You don't see them much nowadays. It's hard to believe they were ever human, isn't it?
Human?
Anyone with susceptibility to the curse, whether they are a saint or a sinner, can succumb to it and become a fiend.
So what does that make you?
Complex. And you?
I'm looking for a greater fiend named Oku. Do you know him?
Why are you looking for him?
I'm going to kill him.
You have no idea what you're up against, do you? If he could be killed, I would have done it myself long ago. He was the one that took my sister from me. It's gonna be a busy night.
You seem to have quite a way with the ladies, my friend. Don't mind me, I'm just passing by. By the way, good luck with that Doku guy. Sounds like you're going to need it. Oh, and I know Rachel quite well. She'll be just fine.
In an age when chaos ruled all, the bloodlines of all the ancient tribes were mixed, and their traits were passed on to the fiends. Then there were the twin fiend sisters, Nike, fiend of destruction, and Ishtaros, fiend of creation. The bloodline they began invited the awakening of an entire new race of fiends. Those who have awakened to become fiends may only find freedom in death. Only in death. Not bad. So what exactly are you? You're much more powerful than just a human.
My strength comes from training, not from some curse in my blood.
Are you so sure of that? I was convinced once that my blood was pure, but then I saw what my pure blood was capable of. I watched as my twin sister was cursed and twisted into a greater fiend. Now I know the path I walk is narrow.
Doku.
He led her down the path of evil, and she succumbed. Fiends are pitiful beings, destined to live forever, slaves to the forces of evil. And there's only one way to release them.
To kill them.
That's why I became a hunter. To kill my sister. To kill Alma. Beneath this monastery, there is...
Was that a fiend as well?
I've never seen one like that before. Something strange. So many of them.
Ayane. Running errands for Mirai again, I see.
The boldness of the fiends is the work of the Dark Dragon. It is a cursed blade, bestowing an unfathomable strength on those wicked enough to wield it. As we speak, its power is growing.
That would explain the stirring in my blood.
Blood. It dominates all humankind, assisted by the power of the fiends.
Ayane, take care of Rachel.
Understood.
Ryu! I've heard there's a hidden path under the monastery that leads to the Emperor's palace. Doku will also be there.
Gamoth. So the Dark Dragon grows in power with each killing.
Of that there is no doubt, My Excellency.
Then I have arrived just in time.
Yes, My Excellency. As always, I provide you with only the most reliable information.
The blade grows stronger and faster than I had expected.
the evil.